হ্যালো, আমাকে শোনা যাচ্ছে?
আমি এখানে 18 লাখ দিয়েছি, বুঝলি? 18 লাখ 95 কিন্তু এর কত পার্সেন্টেজও দেখতে পাচ্ছি না তো। সেটা তো
হুম। ম্যাডাম
Ratnadi, welcome.
Thank you. Good afternoon.
আমাদের স্লাইডটা আমি শেয়ার করেছি এখান থেকে, আপনি বললেন তাহলে বন্ধ করুন, বা আপনি এখান থেকে বন্ধ করে দিতে পারবেন। স্টপ শেয়ার স্ক্রিন।
হ্যাঁ, তুমি কি নামে ঢুকেছো? হ্যাঁ হ্যাঁ, ঢুকুন ঢুকুন ঢুকুন।
স্যার হাত তুলতে বলুন, ওখানে দেখে দেখতে যাবেন সহজে।
তুমি হাতটা তোলো, হ্যান্ড রেজ করো। দিদি স্যার রেডি আছেন তো?
হ্যালো, আমাকে শোনা যাচ্ছে?
হ্যাঁ শোনা যাচ্ছে দিদি, নো প্রবলেম।
হ্যাঁ স্যার রেডি আছেন?
ওকে, আমরা 5 মিনিটের মধ্যে শুরু করব, অন টাইম একদম।
না না, এটা আমি বলছি। হ্যালো পাপিয়া, শোনা যাচ্ছে?
শোনা যাচ্ছে কিন্তু ডিস্টার্ব হচ্ছে তো।
হ্যালো
হ্যাঁ শোনা
হ্যালো
হচ্ছে।
হ্যালো, করবে কারণ ম্যাডামেরটা ওয়েবক্যাম নেই,
ডিস্টার্ব হচ্ছে
আমাদের নেটের জন্য।
তাহলে কি করব?
আমি দেখছি ম্যাডাম তাহলে ছবি ছাড়া জয়েন করুক।
হ্যাঁ তাই করতে বলো।
মানে আমার মোবাইল এটা, ঠিক আছে? আমারটাতে শোনা যাচ্ছে না?
হ্যাঁ এবার শোনা যাচ্ছে।
ম্যাডামকে দেখা যাবে, এটার প্রচন্ড অসুবিধা, কেটে কেটে যায়। শুনতে পাচ্ছ? এটাতে বলো, আপনি এটাতে। না না, সেটা আমি দিচ্ছি, কিন্তু আমি বুঝতে পারছি না যে একটু সবাই চুপ করো। ম্যাডামকে প্যানেলিস্ট, ম্যাডামের মেলে আছে প্যানেলিস্ট, আপনি এটা দেখুন তো দিদি। ম্যাডাম আপনি একটু বলুন, আপনার শোনা যাচ্ছে?
হ্যালো, ক্যান ইউ হিয়ার মি? হ্যালো, ক্যান ইউ অল হিয়ার মি? Yes, yes. Can you see me?
No.
Okay, okay. Hello, নমস্কার, আমি অ্যাকচুয়ালি কলেজে, সেই জন্য I have my নমস্কার। ডক্টর চক্রবর্তী কি আছেন? আমাদের এসেছেন, এখানে আসেননি এখন। আচ্ছা আচ্ছা আচ্ছা, ওকে।
আমি যদি সব ভিডিওতে করি তাহলে দেখা যাচ্ছে না।
হ্যালো,
কিন্তু আমাকে দেখা যাচ্ছে না যখন ঢুকলাম। হ্যালো, আমাকে শোনা যাচ্ছে? হ্যালো, ঠিক আছে ঠিক আছে।
হ্যালো, হ্যাঁ শোনা যাচ্ছে, দেখা যাচ্ছে? হ্যাঁ, ওকে।
You are visible and you are audible.
Okay. হ্যাঁ,
Ratna di, am I audible? Ratna,
আমি আছি।
no, no, am I audible? Someone is asking. আছি, আমরা আছি, হ্যাঁ। Okay, shall I take it up? Shall I take it up? Dr. Chakravarty is also here. Hello.
ready?
Hello. দেখা যাচ্ছে তো। এটা অন্য কারো। This is not ours. আমরা তো দেখা যাচ্ছে। হ্যাঁ, 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 ঠিক আছে, ঠিক আছে। Should we start? Should we start? আমরা কি শুরু করব?
Yes, sir is coming within 2 minutes, please.
Okay, okay, all right, all right. Yeah, sure. I can see Bijoy now. I can see Bijoy. Bijoy ke dekhte pacchi na. I'm never used for, when I used to do this phone থেকে কোনোদিন করিনি তো ওই জন্য একটু আমি একদম পারতে পারি না একদম না। I'm so not used to it. আর একটা কি বলতো? হ্যাঁ, হ্যাঁ, I know, but the thing is that ফোনে না কেউ উল্টো দিক থেকে দেখায়, মানে আমি বাঁ দিকে ঘোরালে ডান দিক যায়। It's like আমি ভীষণ so lost. ল্যাপটপে সব থেকে ভালো।
Good afternoon, sir.
Good afternoon, sir. This is Shujai. Can you hear me?
Hello. Namaskar, doctor. না শুনতে পাচ্ছেন না, শুনতে পাচ্ছেন না, মানে উনি স্টাক হয়ে গেছেন, মানে হ্যালো, আপনি শুনতে পাচ্ছেন ডক্টর চক্রবর্তী? ও মিউট হয়ে গেছে।
Raja, please unmute.
হ্যালো
মিউট হয়ে আছে।
ডক্টর চক্রবর্তী, আপনি একটু আনমিউট করুন। ডক্টর চক্রবর্তী, আপনি কি শুনতে পাচ্ছেন?
একটু ফোনে বলে দিন একটু আনমিউট করার জন্য।
হ্যাঁ রাজা আনমিউট করো।
Okay. So we respect.
স্লাইডটা আমি দিচ্ছি, হ্যাঁ স্লাইডটা দিয়ে তো স্লাইডটা 1 minute.
Okay. Sir, good afternoon. This is Shujai.
হ্যাঁ হ্যাঁ
Shujai বলছি।
হ্যাঁ
শুরু করি আপনার
হ্যাঁ নিশ্চয়ই শুরু করো, দেরি হয়ে গেছে।
So good afternoon to all.
গুড আফটারনুন।
Webinar on Cytogenetics and Genomics Research Unit, Department of and also
Louder, little louder. Voice is not coming very clear way.
this webinar is something special to us because we have a legendary person.
Shujai, I think your voice is it's something is happening. It's breaking up. Your voice is breaking up. We can't hear you.
Voice is not very clear, I'm sorry. Make it a bit more clearer.
Okay.
Can you do that?
Hello.
Hello, voice is not very clear.
Yes, please carry on. Shujai, bolo.
Okay. So today we have a personality, Dr. Baidyanath Chakravarty, and also very eminent scientist and embryologist, Dr. Ratna Chatterjee from Institute of Reproductive Medicine, Calcutta. Dr. B.N. Chakravarty is a world-famous growing personality in the field of medical reproductive medicine. He does any formal introduction. So, I have a long time arranged such a talk for Sir since I started working with him as a collaborator geneticist in the area of male infertility research. But this pandemic gave us opportunity to have Dr. Chakravarty within this seminar as a speaker, and we all are eagerly waiting to hear from his end.
Shujai, কথাই শোনা যাচ্ছে না।
Professor Dr. Ruma Bhattacharya to introduce formally Dr. B. Chakravarty to the audience, also to chair over to Dr. Bhattacharya. Dr. Bhattacharya.
Yes. Thank you, Shujai. Thank you, Shujai. প্রথমেই আমি একটা নমস্কার জানাচ্ছি ডক্টর চক্রবর্তীকে and also Ratnaboli madam ke. Dr. Chakravarty, we have heard so much about you. We have read so much about you, and I think none of us can really introduce you, because you are a very renowned person, and everyone has heard about your work. pioneering work. So on behalf of Bijoy Krishna Girls College family, I would like to welcome you very much. And I would also take this opportunity to thank you. My heartfelt thanks to you, Dr. Chakravarty, for joining us today here. I know your time is very precious, and still you have given us this time to listen to you, to hear you. So we are very thankful, sir. And I know, just the little bit that I can say is that your work in the sphere of reproductive medicine is something that has helped so many couples, so many families are happy today because of you, that their thankfulness, perhaps we should, in this webinar, perhaps we should be thanking you for their happiness. And really, Dr. Chakravarty, I consider myself to be very privileged.
I think I can start now.
Today
Good afternoon.
in front of you, talking to you, and অনেক অ্যাওয়ার্ড পেয়েছেন স্যার আপনি এবং আপনার কাজের সত্যি। হ্যালো, হ্যাঁ, থ্যাংক ইউ। Can you hear us? Can you hear me?
Yes, yes, I can hear. Yeah. Should I start?
Thank you. And Ratna madam, also a big welcome today. Once again, on behalf of Bijoy Krishna Girls College, Howrah, and নমস্কার। আমরা খুবই উদ্গ্রীব হয়ে আছি আপনাদের কাছ থেকে সব জানার জন্য, শোনার জন্য। আমাদের স্টুডেন্টরা, আমি জানি, they will be very much benefited. So thank you. So over to Sujoy. Please carry on, Sujoy. Thank you.
With your permission, ma'am, sir, আপনি এবার শুরু করুন।
Thank you very much, Bijoy, and my student Ratna.
আপনি শুরু করুন, স্যার।
I'm very at the outset. I must apologize for the few minutes delay because of my commitment towards my hospital and towards my patient. Anyway, at the outset, I also am very much privileged, not only privileged but honored as well, because I, after a long time, speaking on behalf or in context with my mother university, from where I started my career. And still I remember that from Patna University in Bihar, when I came to Calcutta University, I was so much delighted that it is my motherland, so I must do something for my motherland as well. Anyway, without wasting any time, let us go to the top topic. And it is a topic which is very favorite to Bijoy, I know, and many scientists, not only in the medical curriculum or medical field, but also in the general scientific field as well. Now, reactive oxygen species is a new terminology which has been built up over the last few decades. And we hear a lot about you in connection with your male infertility. Now, what we talk about reactive oxygen species is that, semen analysis, we do the semen analysis in male infertility, but that's not enough. All of you know about that. Semen analysis undoubtedly plays a central role in the laboratory diagnosis of male partner infertility. Unfortunately, it does not provide accurate data about fertilizing capability of spermatozoa. 30% of all patients with normal-looking sperm have abnormal sperm function. By this time, you must be knowing it. They look normal, and so far as the quantitative point of view, but from qualitative point of view, I think they are not fully functional. One of the facts which adversely affect physiological function of normal appearing sperm is reactive oxygen species, the topic on which I'll be discussing for a few minutes, which is a few minutes ahead. During the last 2 decades, association of RWS with human infertility has created significant interest. Now, what is RWS? RWS is our reactive oxygen species, is an oxygen metabolite. Surprisingly, we survive on oxygen, and at the same time, oxygen metabolites is killing us day by day or inch by inch. How do they do that? This can modify cell function. By modifying our cell function, it can kill us, I mean, quite slowly, but gradually, it's slow poisoning, as you can see. ROS includes molecules like hydrogen peroxide, ions like hypochlorite ion, radicals like hydroxyl radical, and superoxide anion. This is both an ion and radical. As I said, radical, and this is the most important component of that metabolite. I said about three metabolites, like the ion molecule and the radical. And the radical is the most devastating, I mean, radical, which kills the cells every minute, every inch, day by day. Now you can see over here, there's the three metabolites are there. And of these, you can see I have circled that radical. It is the most devastating, or rather more aggressive, most aggressive radical, or the metabolite, which by metabolism, the oxygen can produce and can kill itself. And what is radical? A radical, I have already said, also called the free radical, is the most aggressive component. These free radicals are very violent and may damage a healthy cell. How do they damage a healthy cell? And what is the mechanism behind it? Free radicals are atoms or molecules with one or more unpaired electrons. They are highly aggressive, and they seek or try to find another electron with which they want to bind and pair and can survive. Otherwise, they will also die. Now, how do they damage a healthy cell? In the process of attempting to bind, they attack a healthy cell in order to extract one electron with which they can bind or they want to bind. In this way, they modify or damage many different types of molecules, including proteins, lipids, and nucleic acid, the constituents of a healthy cell. In summary, they damage a healthy cell. And why they are more prone to damage a spermatozoal cell? A small cell, no doubt, in the body, the whole body of a male mankind or the human being, but they want to damage that particular cell, or they are damaged easily because the constituent of a spermatozoa contains some weak lipids which are damaged, and therefore the entire function of the cell is damaged. And as you know, in the beginning, I must say, the spermatozoa has got 4 components, and these components are head, neck, and the midpiece, which contains the mitochondria and the tail. All of these have got different functions, but the peculiarity or the unique behavior of the spermatozoa is not to perform its physiological function in the host itself, but in some other person, that is the woman. That is, the fertilizing capability are all hidden in these 4 components of the spermatozoa. Head, as you know, contains the nucleus, and the nucleus is the fertilizing, most important fertilizing component. The others are helping. The neck contains the centromere or centrosome. They produce that molecule. They produce that mitochondrial. Centriole will produce a pathway or ropeway by which the 2 nuclei will develop, will unite. I'll come to that later on. And the midpiece is the most important one. This mid segment contains the mitochondria. It performs function, metabolism, respiration, and locomotion. The last one is the tail, which contains the dynein, and it is the proprietary force which is gained through this dynein in the tail. So, in summary, that is the way. How do they damage? I was talking about that. Sorry, I'm deviated. In the process of attempting to bind, they attack a healthy cell in order to extract one electron which they want to bind. In this way, they modify or damage many different types of molecules. I have said. In summary, they damage a healthy cell. Now, how sperm cells are affected? As I have already said, this is primarily because a weak biochemical constituents in the sperm cell membrane. One is very rich in polyunsaturated fatty acid, which is known as PUFA, and the sperm head, it has got a central nucleus, less cytoplasm, more nucleus, DNA more. And this nucleus is covered by 5 membranes. One is the plasma membrane, outer acrosomal layer, acrosomal sac, inner acrosomal membrane, and the nuclear membrane. So these membranes are all content of the polyunsaturated fatty acid. Now, this polyunsaturated fatty acid can produce ROS, reactive oxygen species. But ROS can be physiological also, not always pathological. So some amount of ROS is necessary for the physiological function of spermatozoa, and that is the activation. And you know, the sperm goes upwards along the uterine cavity. When it goes upwards, it becomes hyperactivated by some micromolecules in the uterine fluid and in the periovulatory period. And these micromolecules makes the sperm hyperactive. And unless the sperm is hyperactive, it cannot perform its next function, that is the acrosome reaction. Unless acrosome reaction is there, the sperm head cannot penetrate the zona pellucida and go into the perivitalized space to fertilize and oocyte or ooplasm. For the sequential, effect of the spermatozoa, capacitation is a very, very important step, and that is performed by your ROS. But ROS must be in a limited amount, not hyper ROS, not hypo ROS. Hypo ROS will fail to produce that capacitation activity, and therefore the subsequent functions of the spermatozoa will be lost. There's a reason physiological level of ROS generated by healthy spermatozoa is essential for sperm function, and that is after hyperactivation, like hyperactivation, capacitation, and fusion. After that, the sperm pronuclear, the sperm head will fuse with the nuclear membrane, and after fusion, acrosome reaction will occur, and that will degenerate the outer layer of the oocyte, and that the sperm can penetrate into the ooplasm. Another unique characteristic of sperm is less cytoplasm at that particular area, head. And another unique characteristic of sperm is less cytoplasm in the head, and thus have a weak defense mechanism against high ROS level. Excess ROS generation is detrimental for sperm integrity and may cause ultimately a cell death, and that will lead to either oligospermia or agispermia. That is, I have said the PUFA is less, polyunsaturated fatty acid is less, cytoplasm is less, and that is one of the reasons why more ROS can form or generate by some other material which will come in a middle. And one of the many immature sperms are there, many morphological abnormal sperms are there, many lymphocytes are there. They're all ROS generating material, and they will cause ROS and they will gradually damage the sperm cell one by one. Now, in addition, the sperm head nuclear DNA and midpiece mitochondrial DNA are also affected, and the damage affects sperm motility, vitality, and reduced fertilizing potential. I said the three functions of the midpiece. One is the locomotion, the second is the metabolism, and third one is the respiration. And as they are affected, the vitality of the sperm is lost, and therefore the energy of the sperm is affected, and therefore we have the drug which is also known as energizers. In summary, adverse ROS affects spermatozoa. Our affections of spermatozoa consist of impaired motility and fusion events, and deficient fertilizing capacity potential due to DNA damage, and the increased apoptosis. Cell death occurs ultimately in necrosis, and before necrosis, there are two stages of apoptosis. The sperm does not, ROS does not do the necrosis, but it does the apoptosis. And ultimately, when the sperm reaches the oocyte, well, the vitality is already very much reduced because of apoptosis 1 and apoptosis 2. So, common causes of generation. And what is the common cause of generation of ROS in male reproductive tract? The local causes, why they are formed in the reproductive tract? There are local and general causes. The local causes are three groups. One is the presence of many, as I have already said, presence of many immature and abnormal sperm cells in the abnormal sperm cells in the ejaculate, and dysfunctional mitochondria. And mitochondria, from that point of view, energy producer, energy generator. Mitochondria is a very, very important segment of the sperm. I repeat again, 4 segments: head, neck, and the midpiece and the tail. The midpiece is very vital. So for energy content of the spermatozoa is concerned. The next one is leukocytospermia. Activated leukocyte generate 1,000-fold more ROS, and the second one is the varicose, local heat production, and that will produce reactive oxygen species. The third one, this is man-made damage, and that is man-created damage. Rather, when we are preparing the sperm for any type of assisted reproductive technology, we have to centrifuge the sperm, density gradient or normal, high speed up. There are other methods of centrifugation as well, but in all cases, we have to centrifuge. The centrifuge, although we do at 1000 RPM, but their speed limit can always be variable, and that may cause or create ROS in the sperm. Alter temperature and chemical composition of the media in which the sperms are included. So these are all the causes, I mean local causes, in the testes, in the seminal plasma. So they are local causes of generation of reactive oxygen. And the general causes, all you know about that, it produces hypoxia, that is a cardiovascular disease, oxygen saturation might be low, and therefore the local temperature might vary, and that may cause damage to the sperm cell. Diabetes, cancer. The lifestyle between the two, cigarette smoking, excess alcohol, I will blame more cigarette smoking than excess alcohol. Excess alcohol has got a part of beneficial effect to the body, but cigarette smoking can cause damage to all parts of the body, including heart, lungs, kidney, liver, etc. Everywhere there will be damage. There is no, there is no good beneficial effect of your alcohol. Now, as they are producing this, ROS, reactive oxygen species, their enemies are there, but protective forces are always there. Defense is always necessary. And defensive effects are produced by the sperm itself, the cell itself, and also in the media in which the sperm is conveyed or carried. It's a seminal plasma. Now, these antioxidants, they are known as they are not as pro-oxidant. ROS producing, that's the leukocyte or immature sperm, morphologically abnormal sperm. So they are pro-oxidant, they are producing oxidants. And these are antioxidants fighting against those pro-oxidants, naturally produce pro-oxidant. And that will be intracellular on sperm membrane and extracellular in seminal plasma. Sperm membrane and mitochondria both contain some antioxidant, and these are vitamin C, uric acid, alpha tocopherol, etc. On mitochondria, superoxide dismutase, glutathione, peroxidase, and catalase. So these are the three types of prominent types of antioxidant that the sperm has got. In the sperm cell itself and in the extracellular also, seminal plasma, same thing, glutathione, peroxidase, glutathione, peroxidase, superoxide dismutase, albumin, uric acid, vitamin C. So these are all antioxidants, either trying to dispel or scavenge out the ROS that has been generated, or to protect the mitochondrial function to give more energy to the sperm to fight against its ROS. So one of the two things that so this is the physiological protection against the normally generated ROS. Now the balance between the two, antioxidant and the pro-oxidant is known as oxidative stress. The pro-oxidant and antioxidant is oxidative status. What is oxidative status? Is the sperm running, the seminal fluid running with oxidative stress, so the sperm in that seminal plasma will not be able to fertilize. The reasons I have already said, I will repeat again. If the balance is more in favor of ROS, that is the reactive oxygen species generating system, because the leukocytes is the generating system of ROS. Immature spermatocyte, the generating system in ROS. High centrifugation is a generating system of ROS. And those are the pro-oxidant which can help the ROS, that is by scavenging the ROS or by giving more energy to the sperm head, to the sperm membrane, or the sperm, not membrane, there's the midpiece, mitochondria, and to provide more energy and help the sperm to fertilize and oocyte. That's the most important physiological function. There is a fertilizability of the spermatozoa must be maintained till the sperm has reached the oocyte, has penetrated the nuclear membrane, has gone into the ooplasm, the calcium oscillation, and has created the two pronuclei, male pronuclei and the female pronuclei. That is, up to that, there should not be any oxidative stress. The balance is more in favor of ROS generating system compared to ROS scavenging system. The status is known as oxidative stress. Now, this is how, on one side, your ROS scavenging system, it is superoxide dismutase. They are scavenging out as ROS. ROS must be generated, but that must be balanced by the ROS generating system, ROS scavenging system. Superoxide dismutase, catalase, glutathione, peroxidase, vitamin E, vitamin C, vitamin A, and coenzyme 10. So these are the agents by which you're scavenging out the ROS already generated. There are three ways I'll come to that later on. One is always scavenging, the second one is the energized, and the third one is nutrition. So these are the three ways by which you can protect a sperm against the ROS attack. One is scavenging ROS which has already been generated. The second one is your nutraceutical by which you're providing energy. That is the that is the energizers. That is those energizers. The third one is nutritional factor. Now, one thing which you cannot do, I'll come to that later on, is that sperms which are already damaged by ROS cannot be repaired. There is no repairing mechanism. You know by this time that there are three steps in the formation of spermatogenesis. One is from that, I think, what is the proliferation, spermatozoal stem cell proliferation. They're going three ways. They can remain a stem cell, they can proliferate, they can die in the process of division. Because the stem cells are always dividing. And while they are dividing, some of them come towards the spermatozoa, there's a germ cell. And these germ cells, when they're coming, is the first step of the production of the adult spermatozoa is the spermatozoal proliferation. The second step, after some time, they will form the primary spermatocyte, secondary spermatocyte, spermatid, then the round spermatid, and then the elongated spermatid, and adult spermatid. The first step is proliferation. The second step is what is the meiosis. Meiosis is the meiotic division of the nuclear DNA. And half of the nuclear material will be thrown away and half will remain. And the third is the primary spermatocyte. And the second meiotic division is will make it the spermatozoa, secondary spermatocyte, then spermatid, then adult spermatid. The last phase, up to this, if there is some damage or if there is some aneuploidy, that is the division occurs haphazardly, randomly. So when division occurs randomly, well, the reorientation or reorganization will also occur randomly. During this random process, there may be many damage, and that is the reason why so many gametes are abnormal, either oocyte or you call spermatocyte. And we get the spermatocyte, damaged spermatocyte in the ejaculate. But after it has already been damaged, the next stage of differentiation of spermatozoa is spermiogenesis. So first of all, proliferation, meiosis, spermiogenesis. During the stage of proliferation, if there is some mistake in this division or in the differentiation of DNA or in the realignment of the genetic material around the DNA, well, that can be damaged, that can be repaired by the many nucleic acid, many nuclear material, many helping material, repairing material. But after the spermiogenesis, that is when the sperm is taking its adult shape, head created, neck created. So at that stage, well, it cannot be. If some damage has already been performed, so that cannot. That's the reason why spermatid, XY spermatid, is still doubtful, debatable. At least in the round cell Rosling, which you have the ROSNI Rosling, that is still not permitted. It has already been, I think, abandoned. And that is one of the reasons. Because if the damage has already been created or occurred, well, that damage cannot be repaired. So you can just repair up to the stage of, second stage, that is stage of meiosis. But after that, that repair means the repair by giving some, nutraceuticals or some, the scavenging system or by some nutrient material from the later. So that is how it happens. It remains permanent. Now, assessment of this oxidative status. There are methods of measuring the levels because, as I said in the first few slides, that only count and material quantity is not sufficient, quality is more important. And this can be measured by some material, some instrument which we call luminometer. There are methods of measuring the levels of ROS. This offending material can be measured. Upper cutoff, there must be an upper. As I said, some amount of ROS is essential for physiological activation of spermatozoa. Otherwise, the entire process of fertilization of spermatozoa will not be lost. The capacitation, then your capacitation, then piercing the nuclear material, acrosome reaction, that capacitation, acrosome reaction, oxygen, that oxidation, calcium oxidation, all these physiological functions, sequential physical function will be lost. So this cutoff value has been estimated around 75,000 to 1 million. by our team here, because we did not see this cutoff value in the literature. Very confidently, nobody has said about this cutoff value. It is between 75,000 to 1 billion. This is the unit is known as counted photon per minute per 10 million. This cutoff value of ROS provides valuable information in predicting semen quality, fertilizing, and pregnancy outcome in patients undergoing ART. This is estimated by a special instrument known as luminometer. The efficacy of the antioxidant, antioxidant means the protection. There's the pro-oxidant, but there are antioxidants, as I have said. He also measured by another unit, by another type of measurement, and that is known as total antioxidant capacity, or TAC. It can be measured by the luminometer, same instrument. The cutoff value is 1420 molar total equivalent. So that is how we measure the capability or spermatozoal efficiency in the physiological function of fertilization. Now, how can you prevent? As I have already said, there are 3 types of treatment. Either you try to prevent formation of ROS, reactive oxygen species, or try to give some nutrition so that the sperm can fight in spite of presence of ROS, some amount of ROS. That is what is known as the fertilizer. That is the most important one. There's the sperm fertilizer and the sperm scavenger, and the last one is a repair. As I say, we are not yet able to repair the sperm, and the reason I've already said, one, the morphological characteristic of the sperm is already formed, that is the phase of spermiogenesis, not in the phase of proliferation, not in the phase of meiosis, but in the phase of spermiogenesis, when organs are being differentiated, head, neck, mitochondria, there is the body, central part of the tail. At that stage, you can't do it. That's spermiogenesis. Now, correction of varicocele, present treatment of systemic diseases or disorders, avoidance of smoking and alcohol, careful and cautious semen preparation. Because during the time of semen preparation, during the time of vitrification, everywhere there is a possibility of ROS generation. You must be very particular about it. Now, curative treatment, as I have said, as we have already pointed out, removal of excess ROS already generated by free radical scavengers, nutritional supplementation in order to vitalize the sperm to withstand the attack of ROS by mitochondrial energizers. This is known as mitochondrial energizers. Mitochondria has got 3 functions, that is the locomotion, respiration, and metabolism. The conventional treatment currently available consists of antioxidant and nutraceuticals. Now, nutraceuticals, and these are all the work of Dr. Ratna Chatterjee, who has done a thesis, a PhD thesis on this topic. And she has done all this work over the years, 3 or 4 years into about it, and she got the PhD degree in this thesis. And she has these terms I learned from her. There's the nutraceuticals and the antioxidants. All these terms are terminology I knew from her. So depending on the mode of action, these drugs have been classified as free radical scavengers, and not very difficult. It can be done. That is the lycopene, selenium, vitamin E, etc. Mitochondrial energizers, for example, CoQ10, L-carnitine, etc. And nutritional factors, and these are new energizers. We have learned these terms, energizers to the mitochondrial area of the spermatozoa, which provides energy to the spermatozoa. Even if the ROS is there in a limited amount or limited quantity, it can fight back. So these are the things that we have said. Nutritional factors, zinc, arginine, folic acid, vitamin B12. The mitochondrial energizers is the CoQ10, L-carnitine, and acetylene L-carnitine. And the lycopene, as the free radical scavengers are lycopene, selenium, vitamin E, vitamin C, and the glutathione. Now, what are nutraceuticals? We have said the radical scavengers, we have said. Now, what are, and we have said the energizers. These are the energizers, nutraceuticals. Because even if RA is there, it can fight back, because provided they get the good nutrition, and by good nutrition to perform the good function. Every part of the spermatozoa has got some specific function to fertilize an egg. That's the main function. Combination of nutraceutical and pharmaceutical components. Most fruits, vegetables, grains, fish, dairy, and meat products contain several natural components that deliver benefits beyond nutrition. I mean, it has got some pharmaceutical combination. Pharmaceutical combination plus nutrition is known as nutraceuticals. pharmacological, and the nutrition. Examples are lycopene in tomato, omega-3 fatty acid in salmon fish, and saponins in soya. Now, CoQ10 is available in cabbage, carrots, and whole grain oily fish meats. Dose is 60 to 200 milligram per day. L-carnitine is fish, red meat, dairy products, etc. Dose 1 to 3 milligram per day. Lycopene, tomato, red fruits, watermelon, guava, etc. Dose 4,000 microgram per day. Vitamin E and vitamin C, fruits such as citrus fruit, papaya, strawberry, vegetables, dairy, poultry, and meat, etc. So this is a common diet which you every day take, which every man takes every day, but not measuring the amount. But they should not have. And with these nutraceuticals, they can fight back the amount of ROS that is being generated in a normal semen cell. Our observation, that is the Dr. Ratna's observation, and that is, you can say, she has given 6 months as oral supplementation. There's a nutraceutical antioxidant plus oral supplementation diet for 6 months, and we estimated the sperm quality. And there is a ROS, particularly the ROS, and then again estimated after following the withdrawal of the drug after. Not 6 months actually. When she has written 6 months, it is about 3 years. I think she was doing this year work for 3 years. But anyway, she has taken a sample of 6 months and has calculated the amount. And as you can see from this diagram, you can see that everything is consistent and has got significance, excepting 2 facts. One is the leukocyte concentration, the second one is morphology. Although in figure, they show some improvement, but significance has not come. For example, total amount of count has increased from 18 million to 46 million, and the motility from 19% to 35%. But the morphology has remained more or less. A slight increase is there, but not significant. The leukocyte concentration has remained the same. It has not decreased, but at the same time, ROS has decreased. And ROS, you can see here, that is, I think it is 72 to 40. Yes, it has decreased 72 to 41. And TAC has increased definitely. Total antioxidant capacity, that is absorbed, that is the antioxidant. Antioxidant capacity has increased, I think, yes, from 11 to 16. It is also significant. And lipid peroxidase for 75, and the DNA fragmentation has definitely decreased. And all ROS TAC score, this has also improved. So every element or every factor, theoretically or in vitro, it has shown remarkable improvement with this nutraceutical, meaning thereby antioxidant plus nutritional supplement. And when compared with placebo, this placebo they have prepared specially, and they have applied to those, that is 910, and this is something 91 or something, yes, 90. Sample more or less same, and that they have remained more or less same. Total count 21 to 22, motility 18 to 17, and morphology percentage is more or less same. This is also the leukocyte count, ROS, and everything has remained more or less same. Rather, they have deteriorated after your placebo treatment. So, conclusion, what I want to say, or what she wanted to say from our work, that definitely there is a place for antioxidant plus nutraceutical in the treatment of this ROS reactive oxygen species. And the summary of the observation, sperm parameters and reactive oxygen species induced toxic effects were almost similar before and after. Placebo therapy is similar. But after your actual treatment of nutraceutical, it is dissimilar in the sense that they have definite improvement. And therefore, all the parameters except sperm morphology and leukocyte improved considerably after combination therapy of oral combined antioxidant supplementation, diet rich in different antioxidants, and changes in the lifestyle. So they have caused remarkable improvement in the quality of the spermatozoa, which he has examined while doing this work on ROS. So in conclusion, we can say, well, there was some paper who have already shown meta-analysis, some paper share of soil, that is the 2011, reveals that antioxidants and nutraceuticals significantly improve pregnancy and live birth rate in subfertile couples for the ART. Who has got that output semen? I don't say the count is doubtful, but the function is doubtful. I mean, the quality is bad, but the quantity is good. However, large-scale, well-designed, randomized placebo-controlled trials are essential to validate these observations. Thank you very much for your patient hearing. I don't say patient. Thank you very much. And again, yes, I've enjoyed this session. Again, I repeat, this is the university from I made my basic degrees. That is Calcutta University. And I'm lucky enough, not in the, yes, not in the faculty of Calcutta University at the moment. I have been honored, I have been privileged to allow me to talk on behalf of Calcutta University and to the audience of Calcutta University, to the students of Calcutta University, to the teachers of Calcutta University, to the faculties of Calcutta. Thank you very much.
Thank you, sir. We're also honored to have you among us. This is our long time desire that we should listen from your end. And I really, it's a very, very informative and nice presentations. And we learned a lot. I think all the attendees and participants have learned a lot. And now it's a time to take questions. We have 1 and 2 questions. If you give permission, then may I ask from the participants?
Yeah, no.
Yes, yes, why not?
Okay,
Why
sir.
not? Every lecture is questionable.
Yeah, I am taking
And
on.
the lecture goes unquestionable, then it is not a lecture. That means everybody was sleeping at that time.
Privilege to hear from you. This is our fortune that you are there to answer all those questions from our end. So I so from is
Unquestionable, it is unlistenable. শুনতে পাচ্ছি না?
শোনা যাচ্ছে না?
Now it is audible.
Is it audible?
Yeah, yeah. Now it is audible.
Now it is partly audible.
Okay.
Yes, Shurita will help me.
Does luminometer works on the same principle as spectropluorimeter?
Luminometer and spectrophotometer works in the same.
Works on the same principle as spectropluorimeter.
I mean, whether their values collected from the spectrophotometer and the luminometer are the same or not, that you want to know. I can, that part, I mean, Ratna can say. That she can say, because she has done the work herself in the laboratory. I do enter, but not so frequently. And it is Ratna who is doing this thing. But I think they're not same. No?
They are not
Yes,
different.
luminometer is much preferable, I think.
Yes, it
Yes.
is.
And that luminometer, we borrowed, to be very frankly, is not our luminometer. We borrowed it from somebody. That's from Kharagpur IIT. And somebody was doing a thesis also before Ratna. And at that time, this cutoff point, they devised this cutoff point. And we have presented, I think we have published this paper also in human reproduction. And
Yes.
that is doing the follicular fluid harvest. At that time, we were doing the follicular fluid harvest. So this luminometer was brought at that time, and we have already sent it back, given them back. But I think the they are the values represented in the 2 instruments that you have said, luminometer and spectrophotometer.
It is.
So are they same or not, that I can't.
Yes, sir. It is actually the chemiluminescence assay by luminometer. We need one solution, luminol.
Dr. Butcher,
The wavelength of light. That is different.
yes, she has answered it.
Next question is, in case of curative treatment of ROS, how the removal of excess ROS generated by free radical scavengers is performed?
Repeat
Yes,
the
please
question.
repeat the question and a bit more clearly.
in case of curative treatment of ROS, how the removal of excess ROS generated by free radical scavenger is performed.
When we do the treatment,
Antioxidants.
ROS. Scavengers, yes, scavengers are there. There are specific drugs which can scavenge out. I said that only treatment that is available is scavenging, prevention number 1, scavenging number 2, and number 3, vitalizers. These are 3 treatments which are available, practical. And
semen preparation can be
semen preparation is preventive. I'm not talking about preventive, so lifestyle change, then lifestyle change, obesity, reduction of obesity, those treating your cardiological diseases more vigorously. So these are the preventive. I mean, but the preventive is, and as I have said, we are also doing something iatrogenic, doing the centrifugation, and during centrifugation or vitrification, during this there is a possibility of generating ROS. And one of the most important components which we must keep our eyes when we are doing XT at random. And during the process of XT, also there is vitrification can damage or can produce ROS and may damage a healthy spermatozoa. So these things, but that you are looking at the time of injection of spermatozoa, you're looking at the quality of the sperm that you are injecting. And by IMSI, you can improve that technology. How to scan that, how to measure it. So that is the thing. So these are the ways by which you can prevent, the production of ROS. And already ROS produced, you can to a certain extent, not always, can scavenge out, can just remove ROS. But you cannot repair. Once it is damaged, you cannot repair. That's the only thing that I have said, highlighted. Because the repair mechanism is beyond our control in the case of adult spermatozoa or the spermatozoa which has already reached the stage of spermiogenesis. Even at that stage also, giving a spermatid injection has been prohibited. Or one of the reasons is that the repair mechanism is lost. So that spermatozoa at the stage of spermatid, if it is not a proper spermatid, you cannot inject it. Because all spermatids, you cannot see the integrity of the spermatid. And more so, it is called the round spermatid, that is the law is more aggressive than on the elongated spermatid. Elongated spermatids some are well formed, better formed than your round spermatid. At the stage of round spermatid, you cannot predict that this spermatid will go and produce a normal normal functioning spermatozoa. So that is these are the reasons.
Another question. Nutraceutical has any role regarding sperm morphology betterment?
Nutraceutical have any role in sperm morphology improvement?
Nutraceutical morphology. Morphology is no. Once it is damaged, you cannot repair it. But the morphology, how do you know? At the stage, I said the stages of spermatozoal development, and that is the stage primary spermatocyte, secondary spermatocyte morphology is not yet formed. And at the time of spermiogenesis, well, morphology will be formed. The morphological characteristic will be outlined at the stage of morphology. I'm sorry to say, I don't know any procedure by which that stage can be catched up or can be mapped out by any method. That is the stage of transition stage between secondary spermatocyte and spermiogenesis. Because secondary spermatocyte, after that, the spermiogenesis. And sorry, between the meiosis. The meiosis, after meiosis, the third stage will be spermiogenesis. For example, we are looking for, supposing there's a very practical question, and it is always striking in my mind. If there is any way by which you can do that, supposing a couple gets a deformed child, consecutively deformed child twice or thrice. We have excluded all the methods. I always classify this, abortion or miscarriage into 2 groups. One group is the early miscarriage. Early miscarriage means 7 weeks pregnancy has not reached, and the second group is 7 to 10 weeks miscarriage. So early miscarriage is the default or the fault of the gamete. It's either the spermatozoa or the oocyte. And the late miscarriage is the fault of the deciduum or the endometrium. as the implantation form. Now, 7 to 10 weeks, well, we get many clinical aspects by which you know. The commonest cause of the endometrial receptivity defect is, I think, now currently it is going towards the diabetes. I mean, what we see in the society. So diabetes can have the homocysteine, the homocysteine can cause microthrombi, the microthrombi can cause deficient placental vascularization, and that may interrupt pregnancy, and that pregnancy goes up to 10 weeks. It does not go ahead, but the pregnancy which goes only up to 7 weeks repeatedly, not only once, 3 or 4 times or 5 times, and there must be a defect either in the male gamete or in the female. Unfortunately, there is no way by which you know that pregnancy loss in that particular couple is in the male gamete or in the female gamete. But we have the evidences, clinical evidences, we'll publish that paper, that by giving embryo donation, you can get good pregnancy. But doing randomly sperm DAID, donor sperm insemination, or donated egg, well, you cannot know whether the husband was defective or the wife. So anybody can say by mapping out which gamete was defective from the embryo. Well, this is it may be the reactive of, the pre-implantation genetic diagnosis, but that will not give you the correct type of gamete which was defective. That is the problem. And therefore, we are planning to say that at the moment, current phase of our knowledge, through the embryo, if you know, is the male contribution or the female contribution. Then we can be specific on the donation of the gamete, either male gamete or the female. But until you can provide a good evidence that which gamete was defective for creating a bad type of embryo, because the defect is either at the stage of gametogenesis or at the stage of embryogenesis. Gametogenesis, I said the stages, oocytes has also got the stages. So at which stage, meiosis 1, meiosis 2, or at the time of taking the spermiogenesis, well, which stage is very difficult, beyond our knowledge up till now. So that is a problem, legal problem that we are facing. So are we giving, some medicine we are giving, for example, the progesterone or LMWH, low molecular weight heparin, are you doing justice to that? That we are giving only on presumption, on speculation, not on evidence. There is no evidence to show that. And if somebody can provide evidence, it will be good to know for the clinician to know that this is the way in which you should move in this particular case of recurrent or your sporadic. I mean, there are two types of miscarriage. One is recurrent and the sporadic. Sporadic also 90% unexplained and only 10% explained. And the recurrent 50% unexplained, 50% explained. So there's a big chunk of group of patients where the defect must be in the gamete or must be in the embryo. Whether it is the gamete or the embryo, first of all, we have to know that. And there is a particular type of gamete or the sex of the gamete which you have to do. So that the exact treatment will come after that. Up till that, we are doing the treatment only empirically. So that is about our thought so far as the gametogenesis and embryogenesis in the early stages of implantation. Yeah, first of all, gametogenesis may be defective. Then at the time of fertilization, those pronuclear fusion may be defective. Again, there is exchange 5 times. You know that there are 5 times division of the DNA and 5 times reorientation and reorganization. 2 times male gamete, 2 times female gamete, and 1 time at a pronuclear fusion. And the male pronucleus, female pronucleus, they come opposing in each other. Then the pronuclear membrane unites and fuses and dissolves, and then there is again genital tract infection. Not only prostatitis, gonococcal, any streptococcal, all infections will cause the urinary infection. That the varicocele will cause, the sperm damage. Will cause a more ROS. Local heat, varicocele, trauma, cycling may cause local heat and friction. That may also cause ROS. So all these damages, which may cause damage to the spermatozoa, and because the head of the spermatozoa, remember that one only. There is a plasma membrane, outer acrosomal layer, inner acrosomal layer, and the nuclear membrane. So all these are the covering of the sperm. So our plasma membrane particularly is very rich in weak lipid. Weak lipid means that is, I have already said, the PUFA, polyunsaturated fatty acid. And that is the reason why leukocytosis will cause breakdown. And leukocytes always, leukocyte produces always, but ROS has got a fascination for the weak lipid, and that is, they get there, and therefore early premature exocytosis of the acrosin before the sperm head reaches the donor penis, before the acrosin reaction. Because other than ICSI, but the sperm selection is very important. The best sperm, the most competent sperm will perform all these functions. There's a capacitation, acrosin reaction, then the nuclear membrane fusion, and then the penetration in the ooplasm, and the activation, calcium waves, that is the calcium wave creation. All these are functions of that particular area of the head, and that is being done. But it is essential also, some amount of PUFA must be there, otherwise there will be no capacitation. Without PUFA, capacitation, polyunsaturated fatty acid will help in capacitation, but a balanced amount of PUFA is essential, not an unbalanced, not less, not more. So in all steps of embryology, it's a balanced one. The penetration of the embryo, the trophoblast into the endometrium should be very much balanced, not aggressive, neither very soft. If it is soft, miscarriage. If it is soft, IUGR. If it is soft, preeclampsia. If it is too deep, placenta activity. So there must be one balancing agent, and that is in the form of, as you call it, immunological term, cytokine, interleukin, and that is interleukin 2, I think, some interleukin, and that is balancing the more aggressive and the more soft type of formation, aggressive. The stimulus is coming, the stimulus should not be very aggressive for the endometrium to prepare. Endometrium is preparing, meaning thereby remodeling. Remodeling means what? That is, it is being made soft for the trophoblast to penetrate. But it should not penetrate softly, otherwise it will be miscarriage. Miscarriage will go more, or it will lead to your IUGR, intrauterine growth retardation. But if it is more, to prevent that, there must be a balancing factor. And that balancing factor, as you all know by this time, that during implantation, you require the proteolytic enzyme, you require the anti-proteolytic. The proteolytic enzyme are two. It is the MMP, matrix metalloproteinase, and the plasminogen activator, the prostaglandin. But you require antidotes to take the excessive action. And the antidotes, you know, at the same time, the endometrium only produces PAI, plasminogen activator inhibitor, MMP, TIMP, tissue inhibitor metalloproteinase. The prostaglandin, but there is the COX1, COX2. So all these antidotes are already being prepared to maintain a balance between the two forces. One is the penetrating aggressive, other one is the balancing or defensive. So there must be some defense. So these things are being generated in the time of luteal phase, or which we now say that is the endometrial receptivity phase. And that receptivity, concentration between the two, and these two opposing forces will come on the maximum day of receptivity. That is between day 16 to day 20, or day 20 to day 24. Window of implantation. That is known as window of implantation. And all these, molecular structures will come to a balance on those 4 days on which the embryo can implant nicely in a balance, not in an unbalanced way. If it is implanting in an unbalanced way, but the pregnancy will not stay. Either it will end in abortion or it will end in some catastrophe. So that is the mechanism. And nature has created all the time a balancing factor. A balancing factor, aggressive factor, depending factor are there. But the balancing factor is always there. And during remodeling of the endometrium, the balancing factor is there, which is balancing the stimulus driven by the incoming blastocyst and the defense mechanism that is generated within the endometrium during the phases of endometrial receptivity. most important phase is the endometrial remodeling. Remodeling, that is the phase 3. First of all, apposition, then adhesion, then penetration, then inhibition. The phase of penetration is very important. That is the most important step in the case of implantation. Because that is the phase where the endometrial receptivity will be generated by the stimulus being received from the blastocyst and the preparatory changes that are being made with it. That balance will be created at that particular phase during the phase of penetration. There is first of all, your apposition, adhesion, penetration, inhibition. In the phase of penetration, the balance between the two, one is the pro and the other one is the against. I'm sorry, I'm just overstepping.
Okay, thank you for this excellent presentation and so much information we are getting from your end. I think participants have also received such an enriched session and they are really grateful, and we're all grateful to you for giving us your valuable time. And it's a very long time that we are snatching from your very busy schedule. Thank you once again. And
Thank you, বলছি আপনাকে।
আমাকে thank you বলছি, আমি তো আগেই thank you দিয়ে দিয়েছি। Thank you for inviting me to my mother's university.
Mother
University means this is the basic place where I found my basic knowledge, or what you say, yes, platform, from where I have come to this stage. If it is good for me. It is for this Calcutta University who has created my foundation.
আপনি আজকে বলেছেন এটা আমাদের কাছে একটা বিরাট প্রাপ্তি, আমরা যারা শুনলাম বিরাট প্রাপ্তি।
Thank you. Thank you for this discussion, eloquent, elegant discussion.
Thank you. Okay, so now we have our second presentation by Dr. Ratna Chatterjee. Dr. Ratna Chatterjee is Chief Embryologist of Institute of Reproductive Medicine, Calcutta, and also worked as a research fellow in Cleveland Clinic Foundation, US. Ratna Chatterjee is organizing secretary of ISAR 2018 and organizing chairperson of ACE 2020. She has experience teaching. She is an experienced teacher. She teaches FNB students since 2002, working as a faculty member in the ART training program conducted by IRM since 1990. She is also working as a faculty in ART training program conducted by IRRH since 2003. She has total number of more than 20 international publications in peer-reviewed journal, and also has 6 book chapters contributed in the national textbooks. Currently, she is engaged in the research related to idiopathic infertility and stem cell infusion. And also, this is my pride to say that Dr. Ratna Chatterjee is also my collaborator in the field of research of genetics of male idiopathic infertility in the Bengali population. And so it is a time to hear from Madam. But before I hand it over to Madam, I just want to say to the participant, please stay with us. Following Madam's presentation, there is 3 short presentation of 3 young scientists from our group. And you will enjoy, I hope you will also enjoy those 3 short talks related to infertility. So, Madam, this is up to you. Now you can start.
Yes, thank you, Sujay, for your kind introduction. Good evening, everybody. Respected sir, Dr. B.N. Chakravarty. Actually, the passion, the knowledge, the experience I have gathered till today about infertility, about assisted reproductive technology is from this legendary personality. And I am honored to be here today in the same platform with Sir. Thank you, Sujoy. I really thanks to you for considering me to share my experience with all of you. Please just show the slide. Today my topic is glimpses of ICSI, intracytoplasmic sperm injection, and IMSI, intramorphology selected sperm injection. Actually, it is a treatment option, rather best option in severe male factor infertility and azoospermia. Please show the slide. Yes. But before presenting the topic, I should explain what is ART, or assisted reproductive technique. You know, in certain percentage of infertile couple, the basic guidance, the medicine or the hormonal treatment is not sufficient to achieve conception or pregnancy. For them, we have to assist fertilization outside the body in the laboratory, from the laboratory, through some techniques what are known as assisted reproductive techniques. The most commonly practiced assisted reproductive technique is the in vitro fertilization. You know, here we have to collect both the gametes from both the partner outside the body, fertilize them in the laboratory, and the resultant embryo we have to transfer to the mother's womb to achieve conception. IVF is the best option in bilateral fallopian tube blockage, so that the sperms cannot come close to the oocyte and fertilize it. In polycystic ovarian syndrome, you know, it is a hot topic today, even due to anovulation, dysovulation, bad quality oocyte. Sometimes we have to take the help of in vitro fertilization to achieve success. In endometriosis, where both oocyte quality and endometrial receptivity are compromised, again, in some occasion, we have to take the help of IVF. In unexplained infertility, in mild to moderate male factor infertility, you know, the basic sperm parameters in semen, the sperm count, sperm motility, and sperm morphology, when these are below normal, or when there is some abnormality in the sperm function, it is impossible to achieve pregnancy by in vitro fertilization, especially by in vitro fertilization. Severe male factor infertility or in azoospermia, it is impossible to achieve pregnancy by in vitro fertilization. For this particular group of couple to achieve pregnancy, one assisted reproductive technique known as intracytoplasmic sperm injection was invented. Next one. Next slide. Slide, please. Next one. Yes. Intracytoplasmic sperm injection involves micromanipulation of both the gametes to assist fertilization. Next. Actually, here we have to do human manipulation under the microscope. It represents injection of a single sperm directly into the ooplasm or cytoplasm of an oocyte. But to achieve fertilization, we have to inject an alive sperm into the ooplasm of a metaphase 2 oocyte. That means in a oocyte which has extruded the first polar body. It has been reported that injection into an immature oocyte or metaphase 1 oocyte, the fertilization rate is very poor or rather nil. Next one. Next. Next. It is ICSI under pole scope or Polaroid light. Previous one, yes. It was ICSI under Polaroid light microscope or pole scope. You can see the opaque dot. It is an acrosome reacted sperm head. So it is an alive sperm. Here an alive sperm was injected into the ooplasm of a metaphase 2 oocyte. The first polar is seen at 4 o'clock position. So we can expect fertilization from this injection. The right hand side, there is injecting pipette. The left hand side, there is holding pipette to hold the oocyte. And fertilization was happened in this case. Next one. Next. Next. Next slide, please. Next slide. Next one. Yes. The previous one. Previous one. Yes. The common indication of ICSI, I already explained that severe male factor infertility, when the sperm count is below 1 million per ml, when normally it is 15 million per ml, when sperm motility is below 20%, normally it is 32 to 40%, and when sperm morphology is below 4%, the normal is above %. And in case of azoospermic husband, when we could not find any spermatozoa in his ejaculate. In case of repeated IVF failure, it is mainly due to some defect in the sperm-oocyte interaction. The reason behind abnormality in the zona binding or zona penetration, so that if we directly inject the sperm into the ooplasm, we can bypass the zona defect. And in case of thick zona pellucida, you know that in advanced maternal age, the zona may be thickened. If it is more than 22 microns, it is very difficult for the sperm to enter through this zona into the oocyte, and in that case, ICSI is the better option. And thickened zona, you know, happen when in case prolonged artificial culture, such as when we have to do the oocyte cryopreservation due to some medical emergency or due to some sterility, fertilization prevention, etc., fertilization preservation, etc. So in that case, when we are going to fertilize the oocyte, we have to do the ICSI. Next, and in vitro maturation of the oocyte, when we just recovered the sperm oocyte at immature stage, and we cultured them in the laboratory for 24 to 36 hours. In that case also there is zona thickening, and to achieve we have to take the help of ICSI. Next one. Next slide. Please show the slide. Slide show. Slide. Please sharing the slide.
দিদি দিচ্ছে একটু সিগনালে প্রবলেম হচ্ছে মনে হচ্ছে।
So these are the three main indications. There are so many other indications of ICSI, such as in retrograde ejaculation, in case of after chemotherapy, sperm condition is poor in the sample, etc. We have to do the, we have to take the help of this intracytoplasmic sperm injection. Now, for ICSI, you know, those are the other things that matter more than actual injecting the sperm into the oocyte. It requires a lot of instruments, lot of equipments. These are very costly, such as one inverted microscope and two bilateral micromanipulators fitted with the pipette holder. For a right-handed person, the right side, we need the injecting pipette, and on the left side, we need the holding pipette. Actually, holding pipette fixes the oocyte in a certain position, and with the injecting pipette, we have to select the sperm, and then we have to puncture, and with the help of suction, we have to aspirate the sperm beforehand and then withdraw the suction, and we inject the sperm into the ooplasm of the oocyte. This instrument is very costly, about 20 to 24 lakhs. And, we need the Hoffman modulator that is fitted with the inverted microscope, and it created three-dimensional configuration so that the whole procedure is easy and it prevents injury to the gametes. And Tokai heat plate, that means it maintains the temperature at 37 degrees centigrade. You know, ICSI is a time-consuming procedure, especially requirement before, previous, before, yes, this is. Tokai heat plate actually 37 degrees centigrade temperature. When we do the ICSI, sometimes it takes time. We have to find out the good sperm. We have to inject in a proper way. We have to release the sperm properly. So it takes time. So we have to maintain 37 degrees centigrade. Otherwise, it may damage to the meiotic spindle. You know, meiotic spindle is a plate-like structure on which chromosomal alignment takes place. So if we do injection under lower temperature, it may cause failed fertilization, or there may be no abnormal fertilization. Now we need cellular morphology, good cellular morphology of the sperm. We need proper ultrastructure of oocyte and its component. Moreover, for oocyte and sperm preparation, we need polyvinyl pyrrolidine, hyaluronidase and enzyme. Polyvinyl pyrrolidine to make the sperm motile, because sperms are motile some cases, so it is technically impossible, difficult to catch a motile sperm. Moreover, when we inject a motile sperm within the ooplasm, it may cause the damage of the cytoskeletal system. It may cause damage to the meiotic spindle. And so we have to make the sperm immotile by PVP, polyvinyl pyrrolidine, but it is a system, it is a synthetic plastic. It may not be digested by the oocyte lysosome. That's why nowadays we are using sperm catch that is more friendly for the oocyte. And hyaluronidase just to digest the mucin in between cumulus cell. You know that when you first recover the oocyte and identify, it is almost covered by the cumulus cell. But if it is covered by the cumulus cell, then we cannot identify the metaphase 2 site, because for that we have to check the first polar body extrusion. Moreover, if the oocytes are covered with cumulus, it may block the injecting pipette during injection. So we have to do complete denudation of the oocyte with the help of the enzyme hyaluronidase. Pure sperm or all grade wash, it is required for sperm preparation. Sperm preparation means we have to separate the sperm from the seminal plasma before injection. Then culture media for oocyte and embryo culture, liquid paraffin. You know, liquid paraffin is needed. It is essential. It is an essential item or component. It forms a protective barrier between the outside environment and the gametes inside the media. Moreover, it maintains the temperature, osmolality, and pH of the media so that it creates a stable environment for the gametes. And thirdly, it acts as a catchment area for the VOC, volatile organic compounds, you know, that may liberate at any time within the laboratory, and that is hazardous or detrimental for oocyte and embryo health, cause DNA fragmentation. So that we need all these things for any in vitro fertilization and the instruments and other things for the ICSI. Next. Next, yeah, play, yes. Please, it is an inverted microscope fitted with bilateral micromanipulator, and there is one CRI or Polaroid light microscope or polescope. I am very proud that in our institute, due to the initiative of Sir, we have two polescopes, one in the human lab and one in the animal lab, and we have few paper with this polescope study. Next, yes. Cellular morphology of the sperm. Upper left side, you can see the opaque head. That means acrosome reacted sperm. Sir told just now that for fertilization, the sperm should be capacitated, then it should be acrosome reacted, and then only it can bind zona, penetrate the zona, and then cause calcium oscillation, membrane fusion, etc. The upper right, there are vacuoles in the sperm head. Normally, under inverted microscope, it is not visualized. It can be visualized under high magnification, 6,600 and onwards. And if there are vacuoles in the sperm head, it is usually associated with abnormal DNA. So we should not select the sperm with these vacuoles. And that's why IMSI, I will explain later. Lower one, all of you know probably that it is hallosperm test. The sperm head with big halo means healthy DNA. Sperm head without halo signify fragmented DNA, denatured DNA. So if lot of sperms without halo are present in the sample, so there is high chance of fertilization failure, or Sir told that maybe due to male factor defect and early weeks abortion there may be there. And so we have to select the sperm without vacuole, and that way we can avoid the sperm with abnormal DNA during. And you know that DFI, DNA fragmentation index, clinically significant is 25%. And yeah, actually, there was one question that Sir told that by nutraceuticals, by nutrition supplement, by antioxidant, we can eliminate this morphological abnormality. Next one. Next slide, please. Yes, the ultrastructure of oocyte, the previous one. Yes, the upper right, it is the denuded, healthy, mature oocyte. The ooplasm or cytoplasm is with fine granules homogeneous. There is smooth, round first polar body at about 8 o'clock position, thin zona. It is a very healthy, mature oocyte, and under polescope, there is a opaque structure that is meiotic spindle. In a good quality oocyte, meiotic spindle lie close to the first polar body. And that's why during injection, the point of injection site, that should be far away from the first polar body. The first polar body should be at 6 or 12 o'clock position, and site of injection should be at 3 o'clock position to avoid injury to the meiotic spindle, because under normal inverted microscope, we cannot visualize the meiotic spindle. It is an essential component of the oocyte. Only under polescope, that is very costly, we can visualize the meiotic spindle. The lower two oocyte, these are dysmorphic oocyte. The left one is oocyte with centrally located cytoplasmic granules. Even with IVF and ICSI, the fertilization rate is very poor, and right-hand side, there is vacuoles in the oocyte. Again, if few more vacuoles or large vacuoles are there, that means there is deficiency in the cytoplasm, deficiency in the DNA, and it is very difficult to achieve fertilization. And these are maybe due to advanced age group, due to some aggressive stimulation protocol, due to some vitamin D deficiency, maybe another cause, excessive smoking, alcohol consumption in female partner may be the cause. Next one. Next, yes, these are the components used for ICSI, for oocyte and sperm preparation, with green cap, that is hyaluronidase enzyme for denudation of the oocyte, with blue cup, that is PVP, for making the sperm immotile. And the pink one is the culture media for oocyte and embryo culture, and the white big bottle, that is all grad wash for density gradient sperm preparation. Next one. Next. Yes, the whole ICSI procedure we can divide into 4 sectors. Number 1, for any ERT procedure, we have to handle the gametes. So we have to do the gamete collection. That means oocyte retrieval, sperm recovery or retrieval. Then gamete retrieval, we have to do the injection. It is not possible. We have to do some preparation both in the both gametes, the oocyte preparation and the sperm preparation, then ICSI dish preparation, and then final injection. Next. Next one. Yes, the oocyte retrieval, actually after ovarian stimulation, after ovulation trigger at a certain times, within 34.5 to 36 hours, usually after ovulation trigger, we do the oocyte retrieval under USG guidance. And when sperms are present in ejaculate, we have to collect the semen, then with the help of media, with some procedure, we have to separate the seminal plasma, and we have to recover good number of motile sperm for ICSI. And when there is no sperm in the husband's or male partner's semen, then we have to retrieve the sperm either from epididymis by PESA, percutaneous epididymal sperm aspiration, or from testis by TESE, testicular sperm extraction, or TESA, testicular sperm aspiration. Next one. Next. Yeah, it is a short play, please play. Short journey to the lab, the incubator, the sperm. It is VOC meter to assess the VOC. It is another incubator. It is other incubator because the load is more. And the fourth incubator, it is the laminar airflow, where we have to identify the oocyte. We have to identify or evaluate the embryos. Next one. Next. Yes, you can see the multiple follicles. These are the follicles in the ovary under USG guidance. Please play the, yeah. Yes. You can see that under USG guidance, the follicles are aspirated and the follicular fluid are collected, straw color fluid. It is kept at 37 degrees centigrade in test tube, and immediately the aspirate is checked or monitored under the microscope. As soon as we identify the oocyte coronacumulus complex, we have to pick them up and wash in media 3 to 4 times. Here, the oocyte coronacumulus complex, then it is kept in the mini incubator, the benchtop incubator, in media. And when the pickup is over, we just pick up the oocyte coronacumulus complex and dip in the media. This is a mature oocyte just after retrieval. You can see the corona oocyte complex. And then you know that even we do ovulation trigger in a proper way, even we do the pickup in a proper way, there may be mature oocyte, means there may be cytoplasmic, the nuclear maturation, but cytoplasmic maturation always lagging behind. We have, after retrieval, we have to keep the retrieved oocyte in the incubator for at least 1 to 2 hours for cytoplasmic maturation. Next. Next. Yes. Please. Then oocyte, please play the video. Yes, please play. Yeah. It is the oocyte sperm retrieval in an azoospermic husband. local anesthesia is given with a fine needle. Then multiple puncture is done with the wide bore needle, 18 gauge needle, so that we cannot miss the focus of spermatogenesis. Then seminiferous tubule is extracted and it is kept with the help of a mosquito forceps media. Then under the microscope, we have to tease with the help of the two needles. You can see the motile spermatozoa. You can see the male spermatozoa normal adult spermatozoa. So these are the sperm recovery. And next one, and then it was processed, or sometimes it is kept in the media if the sperm number is less, and we have to recover or aspirate the sperm from the media river in the same dish of the oocyte. Next one. Otherwise, if the number is more, we do the density gradient technique. It is, yeah. Next slide. Next, please. Next one. Yes. So the recovery or collection is over. Now we have to keep the oocyte in the incubator for at least 1 hour, and we have to prepare the sperms. If the number is more, through the swim up technique, or if number is very less, then sometimes we can do the, if the number is more, we can do the discontinuous density gradient technique. If the number of sperms are less, then through the swim up technique. And if it is too less, then sometimes we can keep the sample in the incubator and then directly in a small river of media and just keep the sample over there, and directly from the injecting pipette, we have to just aspirate the sperm and do the injection. Now, one thing, you know that nowadays microfluidic preparation is available. The kit is available in the market. In case of DNA fragmentation, sir told that when there is oxidative stress, if the person having diabetes, malignancy, rheumatoid arthritis, or just from unexplained infertility, severe male factor infertility, idiopathic male factor infertility, there may be chances of oxidative stress, and in that case, DNA fragmentation or DNA damage is very common. So in that case, we can just pass the sperm solution or the semen through the microfluidic kit, and that by that we can avoid the centrifugation. Just now, sir told that unnecessary speed of centrifugation, time of centrifugation, that may create iatrogenic ROS generation, and which may cause more DNA fragmentation. So by microfluidic preparation, we can protect the sperm from the further DNA fragmentation. Next one. Now, oocyte preparation, just I told that just after oocyte recovery, we have to keep the retrieved oocyte in media in the incubator for cytoplasmic maturation for at least 1 hour. Then we have to denude the oocyte completely so that we can diagnose whether there is extrusion of the first polar body or it is in metaphase 2 stage or not, and then incubate till injection. Next one. Next slide. Yes, denudation means we have to expose the oocyte coronacumulus complex in hyaluronidase droplet for 40 to 60 seconds. The hyaluronidase droplet, it is very sensitive. We have to keep in the deep fridge, and the concentration is 80 international unit per ml. And after just thawing, then or keeping at 37 degrees centigrade for cumulus complex in the hyaluronidase droplet, and keep for about 40 to 60 seconds, not less, not more, because too less exposure may cause improper denudation, and too much exposure may cause zona breakdown. And after picking up from the hyaluronides, we have to dip in fertilization media and incubate for about half an hour. During this time, enzymatic treatment will be completed. They digest the mucin in between the cumulus cell and just make the cumulus cell loose. So repeated suck in and suck out with the pipette. Here we need 135 microns denupate, because you know that oocyte corona cumulus complex, the diameter of the oocyte near about 120 to 130 microns. So if the diameter is too less, then it may cause injury to the oocyte. Or if it is too much, then the complete denudation may not occur. Denuded oocytes then dip in the droplet of ICSI dish under liquid paraffin and incubate till injection. Next slide. It is a video. Please play the video. It is a Petri dish. Actually, we do the ICSI to avoid the cost in the lid of the Petri dish. Here, for cleaning of the cumulus or denudation, we are using the bottom part. Otherwise, for ICSI, we need the lid. In the middle, there is hyaluronides droplet, and surrounding the media droplet, it is covered with liquid paraffin, because the media droplet volume is very less. So there may be evaporation of the droplet. So always we have to keep under a layer of liquid paraffin. Now, picking up of the oocyte corona cumulus complex, recovered 2 hours before, and deep in the hyaluronides droplet in the middle, and exposure should be only 40 to 60 seconds. And then we have to pick them up and wash in the peripheral media droplet, just successive droplet, to remove the hyaluronides enzyme completely. In successive droplet, we have to wash the hyaluronides from the oocyte corona cumulus complex. And then we have to pick them up and dip in the fertilization media in a Petri dish and keep in the incubator. After half an hour, we have to denude the enzyme-treated corona cumulus complex, oocyte corona cumulus complex. Now, it should be incubated for half an hour. Now, ICSI dish preparation. You know, ICSI dishes are available, but these are very costly. That's why, to avoid cut short the cost, I use the lid of the Petri dish. And the marking is given 1, 2, 3, 4 to avoid re-injection, because otherwise we may do injection in one oocyte twice. And it is the PVP, polyvinyl pyrrolidine, to make the sperm immotile. In the center, that just 0.2 to 0.8 microns, and it is the sperm solution. For ICSI, usually very less sperms are there, and it should lie close to the PVP so that it can percolate in the PVP solution, and that will make the sperm immotile. So we have to keep in the incubator under a layer of liquid paraffin, and then in the meantime, the enzyme-treated oocyte corona cumulus complex, we have to do repeated aspiration and release with the help of 135 microns pipette. That is called denupate, and that will clear up the cumulus completely, and we will be able to see whether they are at metaphase 2 stage with first polar body extrusion or not. Now, just with the help of the pipette make the oocyte completely denuded. If any cumulus is attached to the oocyte, that may cause block during injection. So after denudation, we have to keep the oocyte, one in each peripheral droplet in the ICSI dish. The central drop is with PVP and sperm solution, and the peripheral droplet containing, each droplet containing one oocyte, one denuded oocyte rather. And then we have to incubate. See, there is complete denudation of the oocyte, and it is at metaphase 2 stage, and under whole scope we can see the meiotic spindle. Next one, please. Next, it is over. Next one. Next, it has done. Yes. Now the injection procedure, again, there are 4 steps: alignment of the pipettes, selection, immobilization and aspiration of the sperm, holding the oocyte, and injection. Next, alignment of the pipettes means the holding and injecting pipette should lie in the same plane. For a right-handed person, on the right-hand side, there is injecting pipette. On the left-hand side, there is holding pipette. Next slide. Next. Yes, you can see. We have to bring in the 2 pipettes in the same plane. Left-hand side, the holding pipette outer diameter is more. It is selection of a morphologically normal sperm, then rolling of the tail end to permeabilize the spermatozoa, and then aspirate with the head first. And it is a denuded oocyte. You can see it is a denuded oocyte, and with the help of the holding pipette, now we are going to hold the oocyte. You know, the first polar body is at 12 o'clock position. There are few sperms, it should lie close to the tip, and it is not a good injection. Just to show you, pierce the at 3 o'clock position and then cross the center, but it almost touches the opposite end. That should not be done. And the sperm, just to show you, the sperm is going slowly to the center. First, we have to aspirate little bit ooplasm and then deposit the ooplasm along with the sperm in the. Now we are just releasing the oocyte. Again, we are aspirating the another sperm, and it is another oocyte, but it is not a good oocyte because first polar body. It is not a smooth, round polar body, and there are a little bit cumulus here. And again, the injecting pipette is taking close to the 3 o'clock position, and smooth injection. Slowly we pass the center, aspirate the ooplasm, release the ooplasm along. The sperm was released, and then withdrawn of the pipette and releasing from the holding pipette. This is the second injection. Actually, it is, you can see under high magnification, it is just after injection. Now, after injection, we have to pick up all injected oocyte and dip in the fertilization or cleavage media. Actually, cleavage media is the media which we use after zygote formation. It is dipped in the cleavage media under a layer of liquid paraffin, and immediate incubation is needed. Next day, within 16 to 18 hours, we have to check the polar body, the PN, pronucleus. It is after 26 hours, 2-cell formation of the same injection, 4-cell embryo after 44, it is 8-cell after 68 hours of injection, and it is a blastocyst the picture is not good. You can see another one. It is almost hatching. These blastocyst formation, till blastocyst formation, you can see after injection. Next one. Next one. Sorry, it is done. Now, there are some troubleshooting area. It is not that smooth. We can divide it into 3: during injection, after injection. Next, before injection. Next one, please. Next. Next slide. Before injection, if we expose in hyaluronidase for less than 30 seconds, there may be inadequate denudation, and that may cause blockage of the needle sometimes during injection, but too prolonged exposure may cause zonolysis. Next one. Next. During injection, if we do the alignment not in a proper way, then during injection, we have to just, our tendency is to just quick alignment, quick attempt to align the pipettes, and it may cause breaking of the needle. The needle are very costly, more than 1,000. So we have to be very careful about that, and improper alignment may cause prolonging of time. Impurities in the sperm droplet or sticky sperm. It is a headache, real headache, because it is sometimes the whole semen is sticky. Even after preparation, the stickiness remains, and there is lot of problem to aspirate the sperm and to release the sperm. Sometimes with the withdrawal of the needle after injection, the sperm may come out. And injection of a motile sperm, I told you that injection of a motile sperm may cause damage to the cytoskeletal system, damage to the meiotic spindle. And sometimes when an oocyte is aged, the zona is non-elastic. Usually after injection, the injection site is closed within 60 seconds of injection. But in a non-elastic oocyte, there may be continuous oozing of the ooplasm and cell death. Next one. Next. It is a kind of just bad injection. Just during injection, we should not touch the opposite end. And here, probably I just, to show you, it is a bad oocyte. Too much media should not be poured in, because that causes bursting of the oocyte. Next one. Yes. Now, after injection, you know that in severe male factor infertility in azoospermia, there is high chance of transmission of genetic anomaly in the offspring, 100-fold more chances of transmission of genetic anomaly. So we have to be very careful about that. And there may be fertilization failure even after a smooth injection. If we do improper permeabilization of the sperm, because I told you that after selecting a normal morphology sperm, we have to roll the sperm at the tail end against the bottom of the petri dish. That is known as permeabilization, because it releases the sperm cytosolic factor. Sir told that the sperm cytosolic factor, it releases the phospholipase C zeta protein. Actually, it is present within the acrosome, and that causes calcium release within the oocyte, and it causes the PN formation, the fusion, etc. So if there is improper permeabilization, the sperm cytosolic factor cannot be released, and vigorous mechanical stimulation by rolling the tail is needed when we retrieve the sperm from the testis or epididymis. It is not necessary for mature sperm when we recover the sperm from the ejaculate. Now, inadequate oocyte activation, again, it may happen in globospermia when there is total absence of the acrosome content, when there is not proper mechanical stimulation of the tail end, and when we don't aspirate the ooplasm and deposit it within the ooplasm properly. And injection of an aged or dysfunctional sperm may cause fertilization failure. Next, yeah. So ICSI, though it is an expensive and practice-oriented technique, it offers the only hope for parenting in severe male factor infertility, but thorough andrological and genetic screening are prerequisites for a healthy outcome, and expertise to carry out micro-assisted fertilization is essential in the laboratory. This is actually for ICSI. Just if you are fine, then I can show you the IMSI part also. It is 4 or 5 slides. If you agree, then I can show the IMSI also.
Yes, ma'am, carry on.
Yeah, then next slide. Yeah, majority of sperms used for ICSI in severe or idiopathic male factor infertility may have head defect, too many vacuoles or large vacuoles in the head, and resulting in impaired function or DNA integrity. Next one. Next. And for that reason, morphologically selected sperm injection came in the field of ART. Identify sperm head defect before ICSI, a procedure known as intra-morphologically selected sperm injection, was invented for better screening under high magnification. Magnification should be more than 6,600 onwards up to 8,000. Next. Motile sperm organellar morphology examination under 6,600 using Nomersky optics detect the vacuoles in the sperm head, and the association between large vacuoles with high DNA fragmentation was observed in so many studies. Next one. Next. Yes, it is a slide. Here, octet was used. You can see the sperm, they are too big. And if you see minutely, majority of the sperm have vacuoles. So in severe male factor infertility, sometimes we are not getting proper result and there is abortion in the early weeks. That may be due to injection with sperm with vacuoles in the head. And it is the injecting pipette. It is too big under high magnification. And just we try to catch the sperm without any vacuole in the head. Almost out of 8, 7 have vacuoles in the head. So gradual approaching towards the sperm without vacuole, and then with head first, it was aspirated. And so many sperms, they are having vacuoles. Vacuolated sperms, they are not good. Associated, maybe associated with nuclear defect, DNA fragmentation. Now, just you can see another aspiration. It is very clear, very attractive and interesting. Yeah, but we should take the sperm near to the tip end so that we should not inject much media into the oocyte. The first polar body at 6 o'clock position crosses the center, now aspirate the ooplasm so that the sperm is retreat back. It is oocyte is aspirated, ooplasm is aspirated. Now release the ooplasm along with the sperm. The sperm is now deposited within the ooplasm, and it was actually IMSI, but it takes long hour, very expensive. But there are a lot of studies. Next slide about in favor of IMSI. According to one meta-analysis, top quality embryo formation, implantation, pregnancy, and miscarriage rate were all in favor of IMSI. In severe male factor infertility, IMSI procedure showed significantly higher implantation rate. Next one. Next one, last slide. Yes. So the first 2 points I already discussed. The third point, IMSI may eliminate the chances of ICSI with sperm having nuclear defect, and thus it improves definitely fertilization and implantation rate. Though expensive and time-consuming procedure, IMSI is the best option in unexplained infertility when the defect may be in the male gamete. And Sir told that in recurrent miscarriage, sometimes the male factor is responsible. These male are known as abortigenic male, and due to some head defect or nuclear defect, there is early habitual abortion. And in idiopathic male infertility and advanced maternal age also, IMSI is the best option. Thank you. Thank you, sir. Thank you.
Thank you, ma'am, for a nice presentation, so informative, and these video graphics are just awesome. I think participants have enriched with those video graphic presentations, and they could have a overall idea how these delicate and sophisticated experimentations are being performed in the laboratory conditions, because all of we studied IVF and all these techniques theoretically. We don't have any idea about the dimensions, about the instrumentations, and how these experiments are really, really tough. But still, your nice presentation has depicted as if when I am in the lecture, I just feel that I am in the laboratory to see all these experiments. So really, I think all the participants had similar experience, similar feeling during the course of this lecture, as if we are in the laboratory and you are demonstrating, and we are the students and you are the teachers, and you are demonstrating everything so nicely that all these things have been clear to us vividly. So thank you, ma'am. And again, a lot of questions, and with your kind permissions, may I ask and may I pick one and few of the questions from this lot of questions. I know that it's very difficult to answer all these questions in these sessions, but few can be taken. So the first question is, what is the current success rate of ICSI?
Yes, it is very difficult to say in single word or sentence. You know that fertilization rate is little bit more than conventional IVF, but not it is high, because it is an invasive procedure. And moreover, here we are selecting the sperm against nature. When we are doing the IVF, the sperm, the most potent sperm, it is selected by nature to enter into the oocyte. But in ICSI, it is me. I am selecting the sperm, and the selection may not be perfect. But still, majority of the laboratory in outside, the success rate is little bit more, about 1 to 2% higher than conventional IVF. But in some percentage of cases, we have no option. We have to do the ICSI, such as in severe male factor infertility, in azoospermic male, have to do ICSI. There is no other option. But for other indication, especially I am doing embryology in SIRS laboratory for last 30 years. I still prefer conventional IVF. Only when there is no other way, I do ICSI.
Okay. Next question is from Professor Arya Mitra. Is there any concept of anti-serum antibody in case of infertility?
Yes, now it is not universally accepted. Before, when we attended or we joined over there in SIRS clinic, we do the MART test, we do the antibody test, and definitely these antibody, these IgG, IgA, IgM, they just covered the surface of the spermatozoa, sperm plasma membrane. That's why they just reduce the motility of the spermatozoa. Moreover, they can occupy the receptor in the zona. That's why zona penetration may be hampered. And that's why when there is lot of this clump in the semen sample, we have checked during any investigation cycle of the couple, we see lot of clumping in the semen sample. We can think about this anti-sperm antibody, and then we have to wash thoroughly to dilute the antibody, and we do the ICSI so that less antibody we can inject, or we can overcome the zona binding, etc. But it is not universally accepted. The clinician, they don't prefer the term now.
Next question is, how can we say the vacuolated sperm head means that contains degraded DNA?
Yes, now small vacuole, one small vacuole, that may not cause any harm. But if it is a big vacuole and if too many vacuoles are there, that means there is deficiency of the nucleus, cytoplasm, etc. And that is usually associated with DNA fragmentation.
Okay. The next question is, if anyone have fibroid in the fundus region of uterus, then it can create problem for implantation, or there is chances for normal implantation?
No, normal, the implantation usually not occur in the fundus. And if it is less than, sir told better, less than 4 centimeter, then we can overlook, or if it is just subserous, submucous, but if it is intramural, if it is submucous, then only it can create sometimes some problem. But if it is in the fundus, usually it do not hamper the implantation.
The next question is, up to which age of female partner the IVF is successfully applied?
Actually, due to advanced stage, lot of problems. There may be unexplained infertility, there may be chromosomal abnormality, genetic abnormality, etc. But in our country, we are getting majority of the patient after 35, and we are having pregnancy even after 45, 46. But definitely due to this chromosomal abnormality after 34 years, 35 years, we should do as early as possible when it was detected that there is no other option than IVF. We should do it earlier.
And the next question is, how abnormal fertilization is recognized during IVF?
Yes. Abnormal fertilization, 2 types. Number 1 is no fertilization. And number 2, abnormal fertilization. Normal fertilization is characterized by 2 pronuclei and the 2 polar body, the first and second polar body. But if there are more than 2 pronuclei, or if the 2 pronuclei are asymmetrical, or they are not properly aligned, or they are not equal in size, or if there are 3 PN triploid or 4 PN, then that is abnormal fertilization, or sometimes there is only 1 PN. That may be due to parthenogenesis, or sometimes that may be due to that we are overlooked the second one due to lot of granules in the oocyte.
Next question, how does fallopian blockage affect infertility?
Fallopian tube is the pathway, so that after intercourse, the sperm, after crossing the cervical mucus, after crossing the uterine fluid, they swim into the fallopian tube, and ovulated oocyte captured by the fimbria, and then it was in the fallopian tube, so that they can come in close vicinity and fertilize each other. But if there is any blockage in the fallopian tube, they cannot unite. So in particularly for that group of patients, IVF is the best option.
And the question is next, what is the probability for introduction of genetic problem in case of assisted reproductive techniques?
Yes, assisted reproductive techniques. Actually, here mostly we do in advanced age group, and there are so many other abnormalities, and that may be there are some genetic abnormality, chromosomal abnormality. Moreover, just we are doing against nature. We are taking the gametes outside. Sometimes it is invasive. Sometimes we are taking prolonged hour. That may cause some problem. Though not genetic, but chromosomal abnormality will be there. And moreover, genetic abnormality may be in normal conception also, but for IVF, we have the option to detect the genetic abnormality, sir told, by the PGT, PGS, etc. And by that, we can eliminate this genetic abnormality and we can transfer the good embryo and implant. So there is a role of IVF when there is repeated congenital abnormality, or when there is repeated implantation failure, or there is repeated pregnancy loss due to some genetic abnormality. Then we can do the IVF. We can select the good one by PGS, by PGT, and transfer the good one.
Okay, ma'am, thank you. Thank you for being with us with this 9 presentation. There are
Sir is with me for throughout the lecture.
so, but I would like to request you and also Sad to stay online, because we have 3 young researchers with their lecture in the short topic of only 10 minutes each.
We will be here, but it is difficult for sir to sit over there for a long time.
So that's why I am requesting, if possible, that will be encouraging for the students. That will be encouraging for the new students and researchers. They are the budding scientists, and they expect that you, like Doyen, both of you are the Doyen in this field. You should listen them and criticize them constructively, so that they could enrich in field.
Knowledge about their study. Okay, I am here.
So I would like to speaker. So our first lecture is going to be presented by Somudra Pal. Somudra is the senior scientist.
Bijoy,
Yeah.
yes, please excuse me.
Yes, sir. Okay. Thank you for being with us.
Because it's the whole day that I have worked.
Yeah, yes.
Yeah.
Sir, thank you, sir. Thank you, sir, for being with me.
That's a big thing for us. This is our.
Okay, it's very kind of you. I have been honored, and it's again I repeat that giving a lecture to my mother university campus is something very prestigious and something very honorable at this age. Thank you very much.
Thank you, sir. I should go to you to meet you in person. Thank you, sir. So now the presentation is going to be delivered by Somudra Pal. He is a research scholar at Cytogenetics and Genomics Research Unit, and Somudra's lecture will concern about the epigenetic risk factors related to male infertility. So it's over to Somudra. Video.
একটা করে দাও। এইটা এখান থেকে শেয়ার তো জানি না শেয়ার করে করা যায় তারপর ভিডিও লাস্ট স্লাইড খোলা আছে দাঁড়া এবার ভিডিওটা অন করতে হবে জুম আগে হ্যাঁ সব ভিডিও বলো এখানে দেখো So I think I'm audible. Good evening, everyone. I am Somudra Pal, a CSIR Junior Research Fellow of Cytogenetics and Genomics Research Unit, University of Calcutta, Department of Zoology. I have been pursuing my PhD work since 2018 on this topic, male infertility. And today I'll be talking about all the aspects of male infertility, like how it was considered in the past and how this problem is evolving through the time being right now we are, and what is its future, how we can conduct various research aspects or various research studies to find out the major causes of male infertility. So what's next? So we know what does it mean. This is a male infertility, and it refers to a man's ability to cause pregnancy to a fertile female. And it can also be defined as if a couple is involved in a unprotected intercourse for more than 1 year, around 2 years, but still unable to have a child, then the couple should opt for infertility treatments. And in some cases, in most of the cases, the male person is responsible for this fact. And as we are talking about male infertility, so let's leap into spermatogenesis, how this occurs. This is primary spermatogonial germ cell, primordial germ cell. It is diploid in nature, and it undergoes meiosis to form type A spermatogonium and type B spermatogonium. Type A spermatogonium is kind of a stem cell that reverts back to the primordial germ cell, and type B spermatogonium progressively enters into meiosis. Then it provides the formation of primary spermatocyte. This primary spermatocyte enters into meiosis 1 to form secondary spermatocyte. So you can see here that the 2N or the diploid spermatocyte is transformed into A0 and like haploid secondary spermatocyte, and thereby it enters into meiosis 2. That means 4 spermatids are formed in this phase. Now, this spermatid to spermatozoa, this progression is termed as spermateliosis or spermiogenesis. It is actually a differentiation process, and in this phase, acrosomal cap formation and flagella formation occurs. You can see here clearly in this picture that these are primary spermatogonial germ cell and primary spermatocyte, secondary spermatocyte at deep stain. Here are the spermatids, and here are the mature sperms at the lumen of the seminiferous tubule. Here are the Sertoli cells that provide the nutrition to the sperms, and here are the Leydig cells that secretes testosterone, which is a very important factor for spermatogenesis. So here are several types of male infertility. The most common type, that is azoospermia, that is no or zero sperm is present in the semen suspension. Total absence of sperm in the semen suspension. It is the most common factor, and for that lots of treatments are carried on in this regard. And the second type is oligospermia. If the sperm count is low than a threshold like 15 million spermatoliter, then the condition is called oligospermia. And this is if the sperm motility is low than a threshold, then it is called asthenospermia. The mortality, when the mortality of the sperms are low, then the sperm is unable to move quickly and efficiently towards the egg. And here if the sperms face some morphological issue during development, then it is termed as teratozoospermia. And you can see clearly that head or tail defects, and that might affect the ability of the sperm to reach the egg and fertilize it. And the last one is aspermia, where no ejaculation usually takes place because the seminal fluid ejection path is blocked here. Let's come to the global scenario of this research. About 48.5 million couples currently are suffering from infertility, of which 20 to 30% are male. This is the most important and astonishing factor because before the 50s or 60s, and still now in some part of our country and in some other third world countries, it is still believed that the females are only responsible for these infertility issues. But now it is proven that no, it's not correct. Males are also equally responsible for the infertility factors. You can see here that 15% of the couples are more prone to male infertility worldwide, whereas in India, in our country, 3.9 to 18.8% couples are affected, highest in Kashmir. And we talk about our state, it's around 2.15% couples. So it's 21% of the cases are diagnosed as male. Another astonishing fact is that among the men of different ages, the younger ones are currently more suffering more, and they are more vulnerable to this kind of infertility issues. You can see here in the A graph that those persons who are whose age is below 25, they are actually more vulnerable to the infertility issues rather than the same age of their female. But after 35, you can see the 35 to, in case of the age group of 35 to 40, the females are much more prone to infertility issues. And in this B graph, you can see here the primary infertility rate and the secondary infertility rate is also higher in case of younger in this regard. So as far as we know about male infertility, so we can figure out some potential risk factors of male infertility here. The most important one, I think, increased age at conception of both of the parents. And nowadays it's a burning issue because I think it's a socio-economic effect, because nowadays people are getting more careeristic, the couples are getting more careeristic, and they dream about a high standard of living. So, and they do just thinking, don't want to take lots of responsibilities of a family. So now they usually don't want to get married in the early age, and they don't really take the children for their family. So after 32 or 35, the infertility regarded issues are much more higher. You can see, we had seen it earlier. So in this case, this factor is very much important. And increased obesity, sedentary lifestyle, addiction of alcohol, tobacco, and drugs, they have a cumulative effect on these infertility issues. Not only that, exposure to harmful chemicals like xenobiotic and radioactive chemicals that can cause DNA damage. If you come into the contact of these chemicals intentionally or unintentionally, then they can do a heavy DNA damage, and that can affect your spermatogenesis process. And not only that, the organic pollutants are very much harmful for in this regard, because it provides bioaccumulation, they accumulates in our reproductive tissue, and if the next generation happens, then it transgenetically transforms to the next generation, and this process carries on. Congenital malformation of reproductive tissues. The most important and most easy example of this is testicular dysgenesis that can also cause infertility. Reduced gene expression of potential sex steroids like testosterone, dihydrotestosterone, androstenedione. In our population, we have seen a lot of samples who have these sex steroids are down-regulated in their body. So the hormonal profile is not well maintained, and there is no balance in the hormonal profile. So we can say gene expression happened there. And last but not the least, that is testicular tumor or testicular teratoma. This kind of tumor is not fatal. You will not die in this type of cancer, but it will take the ability of your fertility. So what I've come to know, there are some statements in this regard that the male germline accumulates more and more mutations, and the rate of accumulation of these mutations are faster than the female ones. It is proven. The sperm cells are more susceptible than eggs to the effect of oxidative stress. I will talk about it later, that how oxidative stress affects our infertility, our fertility factors. And spermatogenesis is a cycle that is extremely complex, and it is very, very vulnerable to endogenous and exogenous stresses. And if you study a person's sample, human semen sample, and if you find that the semen sample is okay, then you can conclude that the entire physiology and metabolism of that particular person is very good. So the human semen can become an important environmental and health sentinel as well. So now I'm talking about the effects of various chemicals. There are lots of chemicals you can see here. There are PCBs, PBDEs, PFAs, and some other organic and inorganic chemicals. And they are commonly termed as, they can be commonly termed as endocrine disruptors, because these pollutants have a more similar structure than with the sex steroid hormones like bisphenol and some of these are a very common example over there. So those chemicals usually gets accumulated in our reproductive tissue, and it eventually blocks the endocrine cascade, and the entire pathway is disrupted. It provides a feedback mechanism so that the entire hormonal endocrine balance is disrupted. Not only that, the genotoxic elements like some heavy metals and metalloids like arsenic, cadmium, nickel, lead, and mercury may also damage DNA structure, either directly or indirectly. By directly means the production of the oxidative radicals or free radicals, or indirectly means the alteration of the enzymes that are responsible for the DNA repair. And this cumulative effect can feature a genetic and epigenetic damage, and that might be transmission of the germline to the offspring. It is so you can see here that a transgenerational effect is also present here. Don't stress the could haves only if it's oxidative. Yes, the oxidative stress, Sir had already talked a lot about that. I'm not going into detail. I'm just speaking about talking about a few oxidative stress molecules. Like you can see here, here is a controlled or balanced oxidative stress metabolism. Like you can see that our own body cells provide produce lots of metabolic oxidative metabolites like free radicals, aroma, reactive oxygen species ROS, and reactive nitrogen species RNS, and etc. etc. And these are eventually neutralized by our body's own system, like catalase action, peroxisome action, etc. But this oxidative stress is balanced with the help of the antioxidants. If the oxidative stress gets disbalanced, if the balance is broken, then it will lead to unnatural apoptosis. This is not programmed cell that you can see here that heavy oxidative stress can trigger the Fas-mediated apoptosis here. It triggers the caspase unnaturally, so the cells, the reproductive cells that were not destined to die early. And in this picture, you can clearly see here that endogenous resources, what Baidyanath sir has already explained, that pericell, leukocyte capacitation, immature spermatozoa, the endogenous resources and exogenous resources cumulatively provide oxidative stress that can alter the physiological roles of sperms like capacitation, acrosomal reaction, hyperactivation, and the pathological roles like lipid peroxidation of the sperm, DNA damage, and apoptosis that can combinedly lead us to the infertility issues that are related to male. Not only physiological stresses, the psychological stresses are also very much important. We are facing the stressors in a daily manner. The stressors are actually accumulating stress in these three important endocrine cascade. One is sympathetic adrenal system, one is hypothalamo-pituitary adrenal system, and another is gonadal inhibitory hormonal system. So when the stressor comes and provides stress, this stress effect directly shows a positive impact to systemic adrenal system and provides a negative impact on hypothalamo-pituitary gonadal axis. This downregulates the hypothalamo-pituitary gonadal axis, and now in this regard, testosterone secretion reduces, testosterone expression reduces, so the arrest of spermatogenesis really occurs here. And not only that, the GnIH secretion is also triggered in the response of stress. So it also decreases the HPG cascade, and the result is infertility issues. So these are the future prospects. Why the epigenetics is so much important? See, the sperm epigenome represents a new frontier in the field of human reproduction as epigenetic mechanisms, because epigenetics is that unexplored area which is still needs to be unveiled, right? And epigenetic studies concern specific gene expression changes without altering any of the DNA sequences. And the nuclear condensation within the spermatozoa is quite different, and it's a most stress-sensitive event. Like 85% of the histones in the sperm DNA are replaced by the protamines that ensures the chromatin remodeling. So if there are any error of the packaging of those proteins or protamines, then there might be a very huge impact in the spermatogenesis process. So we will not get mature sperm for the fertility effect. So this is epigenetics. I will not talk a lot about epigenetics. I'm just giving a basic idea about epigenetics, that it is the study of the heritable changes that other than the DNA sequence. So it will not talk about the DNA mutation, the alteration of the DNA, but it is some other kind of heritable changes that affect the gene activity. Then how it occurs? we can broadly classify this epigenetics into three main parts, like histone modifications, DNA methylations, and microRNAs. So histone modification, if you talk about histone modification, we must know about chromatins. Chromatins are actually a hybrid structure of proteins and DNAs. The proteins are usually histone proteins. Histone proteins are 5 types: H1, H2A, H3, H2B, H3, and H4, among which H2A, H2B, H3, and H4 present in doublet to form a core histone octamer that provides a beads on a string structure, and that provides the basic integrity of forming a chromosome. And H1 is a scaffold protein that provides linker facility. So this histone octamer, or every histone protein, has a specific histone tail. This specific histone tail is responsible for some post-translational modifications here, like acetylation, methylation, phosphorylation, and ubiquitinylation. So if you talk about histone acetylation, the acetyl groups are added in the C terminal of the histone proteins, and it is mediated by HAT, histone acetyl transferase, and the acetyl groups are removed by irreversibly by histone deacetylase. And if the methyl groups are added in the histone tail, then the methylase enzymes are termed as histone methyl transferases. And then acetylation of the histone proteins provide the enhanced rate of gene expression, and suppression of the addition of the methyl groups leads to the suppression of the gene expression. So now let's come to the DNA methylation. DNA methylation usually occurs at the CG-rich region of the DNA, and the CG-rich regions are usually promoter, where the transcription factor and other protein factors and RNA ball binds. So methylation at this particular promoter region provides a modulatory expressional changes at the DNA, and the DNA methylation is maintained by a epigenetic marking, and it is also dependent upon the epigenetic imprinting that is provided by transgenetical. And the last one is microRNAs. The microRNAs are produced within our own body to modulate our own gene expression. They are small non-coding RNAs, and they actually find a specific target. These are mRNAs to suppress or to modulate their expression. So how miRNA is produced? This is a simple structure where you can see that the pre-miRNA is formed within our cell, within the nucleus, and the pre-miRNA is then processed by DROSHA DGCR8 complex. This DGCR8 the name came from the Bijoy syndrome, and in our human, in case of Drosophila, this factor is DROSHA. In our human, it is PASHA. So this DROSHA or PASHA DGCR8 complex provides a cleavage to produce a specific pre-miRNA that is exported to the cytosol from the nucleus via exportin 5 transporter. So after the transportation, this pre-miRNA gets attached to the disat TRBP2 complex, and thereby it performs a cleavage. Then the pre-miRNA transformed into 10 to 12 nucleotide, 10 to 12 BP fragments. Then it gets attached with the TRNC6 complex to form a RISC complex, RNA-induced silencing complex. It has two different domains, Argonaut2 and PV domain. And now it gets specifically attached to the specific mRNA and thereby provides the silencing mechanism. So this is a simple interplay between the epigenetic modifications and miRNA in gene regulation. So you can see that the miRNAs can alter the expression of the DNMTs, TETs, and FTOs. So the DNA methylation status, RNA modification, histone modification status can also be changed by the action of miRNAs. So I am just talking about research work, that is altered microRNA expression in patients with non-obstructive oligospermia. The miRNA expression profile in the testis of non-obstructive azoospermia patients and controls were examined first by miRNA microarrays, and the differences in expression level were verified by quantitative RT-PCR method. And the potential mRNA targets of the miRNA are predicted using bioinformatics approach. So the testicular samples were first obtained from the 5 case individuals, age group is 22 to 30 years, and with non-obstructive azoospermia, and from 5 age-matched healthy controls who had no history of meiotic defects or infertility. So here is the methodology. At first, the patient tissue samples are collected with their consent, and thereby RNA is extracted from the tissue samples. Then RNA isolation is done by using triazole agent. And microRNA expression profiles of testicular tissues from non-obstructive azoospermia patients with controls were generated by applying the mi-arcury locked nucleic acid microarray platform. And the RNA was then dual labeled with dyes that spectrally equivalent to Cy3 or Cy5 dyes. They are fluorophores actually. And this is measured using a mi-arcury array powder labeling kit. Next, the labeled microRNAs are used for hybridization at 56 degrees centigrade overnight using a heat shrunk by hybridization bag. Slides were then scanned, then using a GenePix scanner, and microarray images were analyzed using GenePix Pro 6.0 software. And finally, qRT-PCR is done after the bioinformatic analysis for further validation of the microarray result. So you can see here there is a list of oligonucleotides used in this study. And this is the result. This is a heat map of the differential miRNA expression in testicular tissue samples. These tissue samples is clustered according to the expression profile of 173. There are total 173 miRNA samples, twofold. They differentially expressed miRNAs between 5 non-obstructive patients and 5 controls. So the key color bar, you can see the key color bar here. This color bar indicates the miRNA expression level increased from green towards the red color compared to controls. And the dark color here in this heat map indicates the expression level is close to their controls. This is the hierarchical clustering of the miRNA testicular tissue samples. And these are the summary of these down-regulated miRNAs that appear to be in testicular miRNAs. This is based upon the mean fold changes, which is measured by infertile versus control samples. And this is the validation result. This is the qRT-PCR result. You can see here that the microarray data and the qRT-PCR data side by side. This is the in y-axis. This is the expression values. In case of MIR 491, you can see that in non-NOA samples, the expression level usually rises. In case of MIR 302A, the NOA expression level rises a bit. But in case of MIR 520D, the NOA cases have a down regulation of their expression, and in case of MIR 383, there is a down regulation of their expression. So altered expression of miRNAs in the testis of the patients with NOA was first identified for the first time, suggesting that the apparent miRNA expression may contribute to spermatogenesis arrest in human males. So among the differentially expressed miRNAs, it is interesting to note that several members of miRNA clusters like 17 to 92 and MIR 371 to 373 are downregulated in the NOA testicular tissue, which I have mentioned earlier. And in this study, the low expression of these miRNA clusters may contribute to increased apoptosis to the testis in the testis of patients with NOA, because these miRNAs have a potential role in inhibiting apoptosis. So their downregulation can increase apoptosis rate. And expression of the miRNAs in regions of this particular region, 14q32.31, 19q13.42, and some other markers which are mentioned in this X chromosome map, that could be important in spermatogenesis. So this is the final conclusion of my work. It is miRNA expression in testis were profiled and result were compared between patients and not with NOA and the control men. And azoospermic men with testicular failure have different problems ranging from Sertoli cell-only syndrome to the maturation arrest or hypospermatogenesis. So it can be further validated by the study of recombination based study or the meiosis the study. To study the regulatory roles of differentially expressed mRNAs to the target genes, analysis of their cellular origins in larger infertile population needs to be identified. So I have been the ending of the slide by saying that this is a synergistic effect of genetics, epigenetics, and environment. They are not isolated events. Genetics is all about DNA alterations, and their mutations, and the small nucleotide polymorphisms. The epigenetics is all about CpG and methylation, histone modification. The environment is all about the nutrition, drugs, and exogenous and endogenous stress factors. And their cumulative effect combinedly provides huge effect or huge change, we can say, on our phenotype. So the infertility is not dependent upon a single factor. It is a multifactorial disease. And nowadays it is going to be much more personified. We have to make personified medicine for this particular treatment, because you can see that the epigenetics and the metabolism are at all overall related to each other. And this is social epigenomics. Race and ethnicity determine the social environment and social experiences that affect gene function at various stages of life, and thereby influence the health trajectory and modify disease risk. Now it's come time for an acknowledgement. I'm thankful to Department of Zoology, University of Calcutta, ICMR, Government of India, CSIR, Government of India. So and my physician collaborators and participating families for providing us valuable samples for our research purpose. I am thankful to Dr. Sujay Ghosh, my guide, assistant professor, Department of Zoology, University of Calcutta, for providing me such a nice platform to share my thoughts and my research work in front of you. I am very much thankful to Dr. Baidyanath Chakravarty, Director, Institute of Reproductive Medicine, and Dr. Ratna Chatterjee, ma'am, gynecologist and scientist. I am very much thankful to all of them because they are the collaborators of our research work, and they have always supported our research work. And finally, last but not the least, I am thankful to Cytogenetics and Genomics Research Unit, Department of Zoology, University of Calcutta. This is my lab family. Let's begin the journey. Let's see what happens next. Thank you.
Thank you, Samudra, for nice presentation. There are a lot of questions, but due to time constraints, so we are not taking question right now. Our next speaker is Pranab Palodhi. Pranab will talking about some genetic polymorphisms of Y chromosomes that increases the risk of infertility among the Bengali male. Now it's time to hear from the Pranab. Over to Pranab.
Thank you, sir. Am I audible?
Please, carry on.
Thank you, sir. Well, first of all, a very good evening to all the respected delegates, all the teachers and friends. I am Pranab Palodhi, and I am a research scholar from University of Calcutta. And today, the topic of my presentation is polymorphism. Yes, the topic of my presentation is polymorphisms of TSPY1 gene as risk factor for male infertility. To begin with, I must say that infertility is the inability of the couple to conceive after 2 years of unprotected sexual activity as defined by the World Health Organization. Now, testis-specific protein Y-encoded 1 gene is located at the Y chromosome, and the transcription unit of this gene is 2.8 kb long, consisting of 6 exons, that is the coding regions. Now, this TSPY1 gene is primarily known for its implications in development of gonadoblastoma, testicular and prostate cancers, and it is assumed to be involved in the regulations of the spermatogenesis process. Now, regarding the research work, there are several studies exist in so the existing studies include the sex chromosome copy number variants and the autosomal gene polymorphisms, but studies on the Y chromosome genes and the genes controlling the spermatogenesis process are not that much available. And unfortunately, the genetic screening for male infertility in Indian Bengali population is totally lacking. So the objective of our study was to find out whether there is any association between TSPY1 gene polymorphisms and male infertility in West Bengal. So we have worked with 3 different polymorphisms of TSPY1 gene, first of which is RS135888900 C2T substitution, which is an exonic variant, that is a coding variant, which corresponds to an amino acid change from proline to leucine at the 109th position. The next polymorphic variant is RS1297059-542 G2A substitution. This is also an exonic variant, and this corresponds to an amino acid change from glycine to glutamic acid at the 190th position. And the last polymorphic variant is RS566754-601 G2A substitution. This is an intronic variant, that is a non-coding variant, and hence this does not correspond to any amino acid change. Coming to the materials and methods part, this study is a hospital-based case control study. All the samples were taken from Calcutta and its surroundings. The samples were taken only after containing the full consent of the participating families under the governance of our physician friends, and our study was well supported by the Ethical Clearance Committee of University of Calcutta as well as Institute of Reproductive Medicine, Kolkata. Now, firstly, the idiopathic infertile men, that is, men with unexplained infertility, were diagnosed and their semen samples were collected. Then, the patients with chromosomal abnormalities, varicocele, and cancers were excluded. Here, varicocele is the enlargement of the veins in the scrotum, that is, a skin or pouch which holds the testicles. Then, the azoospermic and the oligozoospermic patient samples were detected by seminograms, and they are included in our study as the case samples. We also included the healthy individuals with proven paternity, and they were included in our study as the healthy controls. And then blood samples and the epidemiological data were collected from the selected individuals. In the next step, we have isolated the genomic DNA from the collected samples. Then we designed specific primers for the TSPY1 gene, and we proceed for polymerase chain reaction for amplifying the required portion. Then we were up for Sanger sequencing and detection of polymorphic variants. And last, we proceed for the in silico and suitable statistical analysis. In the result part, to assess the association between TSPY1 risk alleles and male infertility, in case of the first polymorphic variant, RS135888900 C2T substitution, we took the major allele C as the reference, and the minor allele T showed significant association with male infertility, increasing the risk factors by 2.711 folds. Here, the p-value is 0.0103. In case of the next polymorphic variant, RS1297059-542 G2A substitution, the major allele G was taken as reference, and the minor allele A exhibited heavy correlation with the male infertility with an odd ratio of 1.971, and here the p-value is 0.0216. And in case of the last polymorphic variant, RS566754-601, G2A substitution, we again took the major allele G as the reference, and the minor allele A showed extreme confederacy with male infertility with a fold change of 2.411, and here the p-value comes out to be 0.0051. And the bar graph shows the frequency of the major and minor alleles of the 3 polymorphic variants in case as well as the control samples. And here all the 3 p-value came out to be very significant. In the next step, we detected the plausible damaging effects of all the 3 polymorphic variants using in silico analysis in the context of creation or loss of alternate splice sites on the transcripts. This is a pictorial depiction showing the change in the transcription factor bindings in the reference or the wild type sequences as well as the mutated sequences. And the red boxes indicate those changes in the transcription factor binding sites due to the polymorphism. Then we went for functional predictions of the polymorphic alleles through in silico analysis using different softwares, and all the softwares predicted all our 3 polymorphic variants as probably damaging and deleterious. So in a nutshell, we can conclude that TSPY1 gene polymorphisms increase the risk of spermatogenic failure in Bengali-speaking men, as these polymorphic variants can lead to either complete absence of the sperm from the semen or it can lower the sperm count. Then the minor alleles of the SNPs RS135888900, RS1297059 and RS566754 individually increase the risk of male infertility by 2.711, 1.971 and 2.411 folds respectively. Now, lastly, I must say that these are our primary observations, and these study open the doors for the confirmation through the population-based replicative studies on other ethnic groups with larger samples. Now, I'd like to express my thankfulness to the Department of Zoology, University of Calcutta, Institute of Reproductive Medicine, Kolkata, Indian Council of Medical Research, Government of India, all the physician collaborators and participating families for their continuous support. I also thank my supervisor, Dr. Sujoy Ghosh, Assistant Professor, Department of Zoology, University of Calcutta. I also thank Dr. Baidyanath Chakravarty, Director, Institute of Reproductive Medicine, and Dr. Ratna Chatterjee, Chief Embryologist, Institute of Reproductive Medicine. They are our physician collaborators. And last but not the least, I thank the Cytogenetics and Research and Genomics Research Unit, Department of Zoology, University of Calcutta, for their continuous support and motivation. Thank you.
Thank you, Pranav, for a nice presentation and very brief overview of your work. So again, we have a lot of questions, but due to the time constraints, we are not taking questions right now. And now it is the time for the last topic, last talk by Saurabh Datta from our lab. And Saurabh is going to present another important issues related to male infertility. And without delaying, I'm just carry forward this session to the Saurabh. Saurabh, please, ready for presentation.
এটা ফুল স্ক্রিন কি হচ্ছে এটা না শেয়ার স্ক্রিন স্টার্ট ভিডিওটা পুরো হ্যাঁ হ্যাঁ Good evening, everyone. I am Saurabh Datta, and today I'll be talking about the COVID-19, the current pandemic situation, and which is a conundrum in the context of male infertility. as explained by earlier fellows that what is infertility, I'm not going to in detail. And male infertility comprising almost accounts almost 50% of the total infertility cases, and it's a multifactorial syndrome. And as described about the causes of male infertility, most of the area is still unexplained and other comprising of some other causes. So I'm not going into the details about this. It has already told by the others fellows. The main thing is how my today's discussion about the possibility of developing male factor infertility in the current pandemic context, and what reported so far, and how much we need to be concerned, about those men who are at the reproductive age. The first question is from where this story came from. There is a report in the month of February in this year from Thailand Medical News Journal that a group of scientists said that coronavirus might render certain male patients infertile. What did they actually perform? They screened 146 male patients in a group, and among them 10% had abnormal urogenital functions, and they demonstrated the potent pathogenicity to both renal and testicular tissues with resultant lesions, which is confirmed by immunostaining. And they advised clinicians should pay attention to the risk of testicular lesions of these particular young patients for their fertility. And right after their publication, another group of clinicians come scientists talking about this story and claim that new coronavirus may cause damage to a man's testicles, and they urged that male patients to take fertility tests upon their recovery. So what is known to us? It is already known to us that SARS-CoV-2 may gain their access into the host cell by binding to the ACE2 receptor with higher affinity than reported the previous SARS-CoV-1. And then priming was done by transmembrane protease, serine protease 2 subsequently. And these particular receptors are widely expressed in the lungs, cardiovascular system, gastrointestinal system, kidneys, neurological tissues, and the testes. So there is evidence about the relationship between the members of coronavirus family and orchitis in human. What is orchitis? It's an inflammatory condition of one or both the testicles. So after this, what is unknown about SARS-CoV-2 and its relation to the testicular involvement is largely unknown. What are the objectives? The first thing is ACE2 expression in testicular cells for SARS-CoV-2 and associated pathological findings. Number two is possible mechanism of infection and subsequent impairment in the reproductive system of male. The vulnerable age group, impact of SARS-CoV-2 on male reproductive hormones, and assessment of SARS-CoV-2 in seminal fluids. A publication by Wang et al. In this year, they showed that Sertoli cells and spermatogonial cells are chiefly expressing this particular angiotensin converting enzyme 2 receptor through which the virus can uptake into the cell or into the host cell. And gene ontology analysis from their study, it also showed that genes associated with viral reproduction and transmission were highly enriched in SE2 positive spermatogonia, while the genes which are associated for the procedure of spermatogenesis was downregulated. So, and the reverse is also true for SE2 negative spermatogonia. So from this study, we can find out that the testis is a high risk organ and which is equally vulnerable to SARS-CoV-2 infection that may result in spermatogenic failure, thereby dysfunction of male reproductive system. And the possible pathological findings was published by another journal. Here it is clearly shown that this long arrow indicates the Leydig cells which strongly express the ACE2 receptor, and these are the spermatogonial cells which is negative for ACE2 receptor because
it's very difficult to assess through immunostaining process a particular stage of a receptor, particular stage through which the receptor is associated. And Sertoli cells, these very open arrows indicate the covering of spermatogonia, which is in Sertoli cells, is also positive. So from this finding, it could have been said that testes from COVID-19 patients exhibited significant seminiferous tubular injury, reduced Leydig cells, and these findings can provide evidence-based guidance for sperm donation and inform management strategies to mitigate the possible risk of testicular injury. And there is another interesting story came from this particular publication, that there is a delayed clearance of SARS-CoV-2 in male compared to female patients, just because they showed that there is a distinct high ACE2 expression in testicular cells when comparison to women in their ovarian cells. So from this study, this may also support that higher vulnerability of SARS-CoV-2 mediated impairment in male gonadal functions particularly. And this is the possible way of mechanism, and it is already known that how the virus is being uptaken by the host cell, and after getting infected, the inflammatory pro-inflammatory molecules are upregulated, and thereby several pathophysiological manifestations will come out, like orchitis, as I mentioned. Low level of testosterone is also reported. I will tell it later. Followed by DNA fragmentation that might be happen due to the consumption of antiviral drugs like ribavirin, and followed by, due to huge oxidative stress caused by the pro-inflammatory cytokines, the Sertoli cells disrupted the process of spermatogenesis and thereby formation of abnormal sperm. So the question is, what is the vulnerable age group? A particular publication in Journal of Cellular and Molecular Medicine showed interestingly that there is a testicular expression of ACE2 which is largely age related, and the highest expression recorded in patients aged 30, which is higher than those in their 20s, whereas age group of 60 or above showed lowest level of expression in their testicular cells. And large proportion of these vulnerable males is in their childbearing age, and thus their reproductive ability can be affected. So this might indicate that young male patients are more prone to the testicular damage by COVID-19 in comparison to older patients. What is the effect of SARS-CoV-2 infection in relation to the male reproductive hormones? There are 3 independent studies through which we can conclude about the reproductive hormones related to male. In the first study, they showed that the luteinizing hormone was significantly increased due to infection, but the ratio of testosterone to LH and the ratio of follicle stimulating hormone to LH were dramatically decreased, and the level of testosterone is also decreased in the COVID-19 males. Another study also reported that testosterone levels were negatively related to CRP levels, with low total testosterone levels observed in most severe cases. And another study from third study from Germany also reported the same thing, that is the low testosterone and dihydrotestosterone levels in the COVID-19 patients. So from this finding, it could have been suggested that direct effects of SARS-CoV-2 infection in testicular cells rather than via hypothalamic pituitary testicular axis and hypogonadism might be considered as a risk factor for COVID-19. It's an alarming condition for reproductive aged men who are affected by SARS-CoV-2 infection. And this is the possible way through which the viral uptake directly impaired the process of spermatogenesis through decreased intratesticular testosterone levels. It's a putative mechanism. And last question is that the assessment of seminal fluid, the semen, and the viral load, how it is being related. One study from China showed that in 38 patients who provided semen samples, among them 23 patients had achieved clinical recovery and 15 participants were at the acute stage of infection. Among them, 2 found positive from the recovery patients and 4 found positive from the patients who are at acute stage of infection, and altogether total 6 had results positive for SARS-CoV-2. From this study, it could have been said that the SARS-CoV-2 can be present in the semen of patients with COVID-19 if it could be proved that SARS-CoV-2 can be transmitted sexually in future studies. Sexual transmission might be a critical part of the prevention of transmission, especially considering the fact that SARS-CoV-2 was detected in the semen of recovering patients. And it is not unreasonable to believe that this particular viral load can be found in the semen of affected or not affected or recovered male, because the same thing happened in case of earlier studies involving Ebola virus, Zika virus, herpes simplex virus, and another thing. But subsequently, another study from UK and also from China showed there is no such evidence of SARS-CoV-2 virus in the males recovered from the COVID-19 patients. And this is the study in brief. From this study, this observation also support that it should not be definitely rule out the presence of SARS-CoV-2 in the seminal fluid during an acute infection with severe COVID-19 symptoms. So altogether, it's a take-home message is like, number one, the 4 main testicular cell types showing expression of ACE2 mRNA are the seminiferous duct cells, spermatogonia, Leydig cells, and Sertoli cells. It's a breakthrough revelation in male fertility research. Number 2, male patients are reportedly slightly more affected than women as they possess a larger expression of ACE2 in comparison to women. SARS-CoV-2 can operate via multiple possible mechanisms which may lead to disruption of male reproductive functions. Orchitis, that is the inflammation of the testicles, was one of its accepted health complications that may lead to disruption of the process spermatogenesis, germ cell apoptosis, thereby affecting the semen parameters. Number 4, young male patients are comparatively at higher risk of testicular damage in comparison to older one. Studies reporting the influence of COVID-19 infection on testosterone are contradictory. Still, the hormone, this particular hormone, generally reported to be lower in COVID-19 patients. And in 5 out of 6 studies so far, it has been confirmed that viral mRNA was not detected in seminal fluids of COVID-19 patients. So the potential sexual transmission of SARS-CoV-2 is likely low, but requires further investigation. Therefore, further research is needed to understand the long-term impact of SARS-CoV-2 on male reproductive function, including fertility and testicular endocrine function. So finally, I would like to acknowledge my guide, Dr. Sujoy Ghosh, for his relentless support and guidance and for giving me this opportunity to present this work. I would like to express my sincere gratitude to HOD of the Department of Zoology, University of Calcutta, and equally thankful to our collaborators, Dr. Baidyanath Chakravarty and Ratna Chatterjee, madam, and grateful to all my lab mates, another family from me, for their constant encouragement and support. And this is my family, that is Cytogenetics and Genomics Research Unit. And lastly, thank you for your patience. Thank you.
Thank you, Saurabh, for nice presentation. And this is the end of sessions. Before ending the sessions, I just want to congratulate all of you and convey my heartiest thanks and gratefulness once again to Dr. B.N. Chakravarty and Dr. Ratna Chatterjee. And Didi, you are so kind to us that you stay entire sessions with us. That is definitely is an honor. So these young boys are really anxious to deliver their own work and their reviews and everything. So once again, thank you to all for keeping patience for a long time, and you will have your certificate. And participants, your feedback link is open for another 45 minutes. So you just put your feedback in their respective feedback link and you will get your certificate within a couple of days. Thank you. Have a nice evening to all. Thank you, Didi. Again, thank you. I'm just ending the session. Thank you to all.